Hear me back? Yes, thank you. Good morning. My apologies for starting late. Our committee members are in a bus that is stuck somewhere around Ames. We were waiting for them to be here. Nevertheless, we'll start. Well, thank you all for being here. This is the meat of the event, the two-day science meeting that we'll do, and I'll take you through the event and a little bit about the background of IndiaAlliance, and then we'll have the talks as per the program. IndiaAlliance is a partnership between the Wellcome Trust and DBT. This was a partnership that started in 2008. Our fellowship program started in 2009. And we started with 3 fellowships, early, intermediate, and senior. Along the way, we also introduced a Margdarshi fellowship, and now we complete 10 years this year. There is a plan to go into the next phase for another 5 years. That's something we call phase 2. And in phase 2, along with the fellowships, we also plan to introduce collaborative grants, which we call team science, and want to bring in ways and means to strengthen clinical research in the country. I will talk more about the future of IndiaAlliance at the very end of this event tomorrow evening. Our mission from the beginning really has been to enable biomedical research in India, and we do it through funding and engagement. Funding is important and engagement is equally important, engaging with people, with communities to take this forward. We like to empower our researchers. We try to select the best people who are internationally competitive, and then nurture them to become leaders of the future. We bridge gaps by facilitating engagement between science and society, and develop interventions to meet the demands of the research ecosystem. And of course, we strive for excellence, because without quality, everything else is meaningless. IndiaAlliance has 3 levels of fellowships: early career fellowships, intermediate, and senior fellowships. And the details of these can be seen from our website, so I'm not going to spend time talking about those. Taking stock of what we've done over the last 10 years, we have given 320 awards since May 2018. We have another round of fellowship interviews coming up later in this week. So we will add to this tally maybe another 20 or so fellows, maybe more. Our funding rate over the last 10 years has been roughly 10%. So those of you who are our fellows, who are in this room, you are really the cream of biomedical research in this country. I'm not saying the creamy layer, that has a different connotation in India. You are the cream. We have a pan-India distribution. So far, we have funded people at 93 different institutions in 34 cities. So no matter what people tell you that this is a Delhi, Bangalore, Pune fellowship, it is not. It is a pan-India fellowship program. This event celebrates 10 years of IndiaAlliance. This is the logo that we developed for 10 years. Yesterday, we were honored that the Honorable President was with us to celebrate 10 years of IndiaAlliance. And this is something that his office tweeted yesterday, and we are very honored to get this kind of attention. And the President also honored us by letting us take a photograph of the entire staff with the President, with the Minister, and other dignitaries. And we are really very pleased with this. We actually started yesterday with public talks, which we have initiated with this meeting is called the Explorer Series. Why Explorer Series? Because every scientist is an explorer, every child is an explorer, and we really want scientists, our fellows, to be childlike in their curiosity and in their enthusiasm to move the work forward. We had 4 talks yesterday by 4 of our fellows who really went down to the level of college students and told them about their journeys. And we had this room full of about 500 college students yesterday. It was really a fantastic atmosphere. In your bags, you will also find a volume of Nature India that we commissioned. We take responsibility for all the editorial content of this, and we are very pleased that some of the best people in the world in their areas of research have written in this commemorative issue. I'm sure you will enjoy reading this, and this we will certainly treasure for many, many years to come. So thank you everyone who has contributed to this special issue. In your bags, you will also find a book zine. It's like a cartoon sketchbook about the research that our fellows do. And this is something that we in the office developed with the help of a scientist come artist. And this is something that I hope that you will use to propagate science to society, to young students. We're also bringing out a Nature Inside View. This will be published in the Nature, which is coming out this week. So the Nature which gets published on Thursday, which is released on Friday, would have an inside view telling about our 10th year events. There is Stories of Science, which is an exhibition of images of science, images produced by our fellows, which are on display at Jacaranda Hall, where you will have your tea and your lunches. Please spend time looking at these images, and you, the audience, will judge these images, and we have some prizes for the images. So all of you are experts. You will go out and judge the images. This is a mini website that went online yesterday, which describes all the 10-year events from 2008 till now, what we have done about this event. Thank you very much. I'll stop here and again welcome you to this event. Thank you very much.
Good morning, everyone, and welcome to the second day of celebrating Decade of Discovery. We will now have the keynote lecture by Professor Ashok Venkataraman. Professor Venkataraman is the director of MRC Cancer Unit, University of Cambridge, UK. His laboratory studies chromosomal instability in cancer pathogenesis and treatment. He is associated with IndiaAlliance as a member of the Intermediate and Senior Fellowship Selection Committee member. Ashok, please.
Thank you very much for the introduction. I'm afraid my slides are just coming up, so please bear with me for a minute. Perfect. It's a real pleasure to be here this morning to be speaking to you. And what I'd like to do in the next 30 minutes or so is to share with you some insights from my lab over the last few years concerning the problems that we've been working on for more than 2 decades. But before I start talking about my science, I'd like to also take this opportunity to congratulate Shahid and his colleagues at the IndiaAlliance for their 10-year anniversary. As I think all of us in this audience will agree, it's really been tremendous that the IndiaAlliance has, within a relatively short space of time, been able to make such a wonderful impact on cultivating Indian science, particularly through funding promising young investigators. And it's been a real privilege for me to be associated with the IndiaAlliance in a very small way over the last several years. So with that said, what I'd like to do is to talk about the work of my lab as exemplified in the title, From Cancer Susceptibility Mechanisms Towards Early Intervention in Cancer. So as many of you know, the work that we have been doing in Cambridge over the last 2 decades has really been focused around, if you will, a dream. And that dream is whether in cancer, as in many other chronic diseases, there are opportunities for early intervention. What do I mean by early intervention? For me, the scientific issues are encapsulated in these 3 questions. First, can we understand, before they develop cancer, which individuals amongst the population are most susceptible to the disease? Once the disease begins, can we understand which early lesions are most likely to progress? Because as we well know, not all of them will. And finally, with this information, can one begin to halt progression early, or even potentially in the future to prevent or delay the onset of cancer? And these questions obviously need a good understanding of the conspiracy, if you will, between environmental factors and genetic risk causing susceptibility to cancer, a good understanding of the key molecular and cellular events during early steps in oncogenesis, and finally, enabling technologies to be able to try to take some of this information towards clinical application. My own work started on this problem roughly 2 decades ago, trying to answer or address a simple clinical observation that I came across when I was training in medicine here at the Christian Medical College in Vellore. The observation is that in most common sites of carcinogenesis, very few incipient neoplastic lesions actually progress to invasive malignancy. And this, of course, implies that there must be multiple homeostatic mechanisms, both cell-autonomous as well as non-cell autonomous, whose purpose it is to eliminate or prevent the progression of these early lesions, and mostly they work very well. But sometimes they fail, and that is when incipient malignancies are able to proceed towards invasive disease. So our somewhat simplistic thinking when I started my laboratory off at the MRC, Laboratory of Molecular Biology in Cambridge, was to ask whether looking at families or people with inherited early onset cancer syndromes would give us clues as to what these mechanisms were that prevented cancer. The rationale being that in these individuals, the genetic mutations that conferred early onset cancer predisposition must in turn affect one or more of these homeostatic mechanisms. We were very lucky because at the time we started on this problem, there had been major advances in understanding the genetics of cancer susceptibility, particularly in breast cancer. What I say here applies to most epithelial cancers, although I'll illustrate it with breast as an example. Only about 5% of epithelial cancer cases exhibit a strong familial pattern of inheritance. The majority are sporadic. We now know that germline mutations contribute to high penetrance risk for many of these familial cases, but not all. Even in breast cancer, which is arguably one of the best studied epithelial neoplasia, we only understand about half of the familial risk. Only about half. Can you believe it? After 20 years of study. But what we do know is that certain high penetrance mutations affecting genes like the so-called breast cancer genes, BRCA1 and BRCA2, are important predictors of susceptibility to familial cases of breast and ovarian cancer. And what we now also know is that it's not just germline mutations affecting these genes, but also a number of somatic mutations either affecting these genes or affecting the pathways, the cellular pathways that they work in, that contribute to the majority of sporadic cases of cancer as well. Regarding BRCA1 and BRCA2, which I'm going to speak about now, we know that inheritance of heterozygous mutations, in other words, mutations affecting a single one of the two copies, suffices for predisposition not only to breast cancer, but also to cancers of the ovary, pancreas, prostate, and many other tissues. We know that these are high penetrance mutations in that carriers exhibit a very high cumulative risk of developing these types of cancer by the age of about 70 to 80 years. Interestingly, it's become apparent quite recently that mutations affecting these germline cancer susceptibility alleles may account for a significant fraction of many human cancers. So this is a recent study from Wash University where they looked at over 10,000 cancers of 33 different types. And what you see here is a plotting of the percentage of cancers at various sites that can be accounted for by pathogenic germline variations. And what you see is that mutations affecting the breast cancer genes, BRCA1, BRCA2, a partner of BRCA2 called PALB2, account for a very high frequency, relatively speaking, of cancers of the breast, the ovary, the pancreas, the esophagus. In other words, several relatively common epithelial sites. So what I'd like to share with you over the next 20 to 25 minutes is some of the work that we've done in my lab trying to use these pathogenic germline susceptibility alleles to exemplify opportunities for early intervention in cancer. In other words, through understanding how mutations in these genes, like BRCA2, confer cancer risk, and by understanding the processes involved in carcinogenesis, can we actually begin to devise strategies, at least in this high-risk population, for early detection, risk stratification, and potential early intervention. So to think about some of the recent data from my lab, let me begin by telling you what I suspect every single person in the audience will know, which is the so-called Knudsen two-hit paradigm for tumor suppression. So please bear with me. As I said, I'm sure this is familiar to virtually all of you. The idea is that for germline inheritance of susceptibility alleles for cancer, tumor suppressor alleles, that you inherit a single copy of a mutant allele. The second copy is lost in certain cells. This is called, confusingly, loss of heterozygosity, but it actually means mutations affecting both alleles. And this loss of heterozygosity event, the second hit, if you will, is what promotes carcinogenesis. And when we started our work, it was believed that this LOH event, the loss of that second copy, was vital for carcinogenesis to begin. We now know, through the work of my lab and many others across the world, why this may be the case. We know that when you inactivate BRCA2, both copies of BRCA2 by loss of heterozygosity, cells exhibit spontaneous chromosomal instability. And it's literally 20 years ago to this year when my lab published this work showing that when you knock out both copies of BRCA2 in the mouse germline, you get these grotesque chromosomal abnormalities, both of chromosome structure and number. And you see some of them highlighted by arrows here. Those of you who look at chromosomes will immediately realize that the vast majority of these lesions are of the so-called chromatid type. In other words, there are breaks affecting one of the two sister chromatids, or these grotesque structures called radial chromosomes, triradials or quadriradials. And these are pathognomonic of many cancer susceptibility syndromes, such as, for example, Bloom syndrome. And they led us, when we found this at the time, to suggest that genome instability, in a manner similar to cancer susceptibility syndromes like Bloom's or Fanconi anemia, was what underlay, if you will, carcinogenesis following BRCA2 inactivation. We now know, through the work of many labs, that BRCA2 inactivation disrupts a critical form of repair of DNA lesions. This is DNA repair by homologous recombination, which is a mechanism that mammalian cells use to deal with double-stranded DNA breaks. So when you get a break, which is typically flush-ended, you get nucleases that resect one of those strands, leaving a long overhanging single strand of DNA. BRCA2 then catalyzes reactions that bring to this single-stranded DNA substrate the recombination enzyme RAD51, which is the mammalian homologue of bacterial REC-A, which many of you will be familiar with. And RAD51 forms an ordered helical nucleoprotein filament on the single-stranded DNA, and that structure enables invasion of the homologous duplex, the templated strand, and that leads to the strand exchange events that lead to error-free repair by homologous recombination. And you immediately see, when I describe this process, why you get chromatid-type errors in BRCA2 deficient cells, and you also realize that this process is vital in all dividing cells. It can only work during the S and G2 phases of the cell cycle when you have a perfect templated copy against which to perform the repair. So when BRCA2 is inactivated, this process is gone, and what you're left with are error-prone processes that repair double-strand DNA breaks by mutagenic events leading to mutations, and we think carcinogenesis. We now understand how BRCA2 promotes RAD51-mediated homologous DNA recombination in some detail. And I'm not going to take you through all of the work, but just highlight a few of the steps that we have identified in my lab. So this is BRCA2, and what you see here is that it's a ginormous protein. My postdocs still curse me because it's 3,500 amino acids long. No one can purify it as a single isolated protein. It's extremely difficult to do biochemistry or structural work with, and we, like many others, have therefore been looking at pieces of the protein rather than the full-length molecule. So one of the first structures that we solved with Luca Pellegrini in the Department of Biochemistry at Cambridge, going on over a decade ago, was the structure between these 8 repeated sequences, the so-called BRC repeats of BRCA2, and a single molecule, the core catalytic domain of RAD51. The point is that the interaction between the BRC repeat and RAD51 actually blocks the capability of RAD51 to oligomerize on DNA substrates. And if you'd been listening to what I said, you'll realize that oligomerization is a fundamental characteristic of RAD51, which enables it to form this ordered helical nucleoprotein filament. So what is a protein like BRCA2 that promotes RAD51 function doing by antagonizing, apparently, the ability of RAD51 to oligomerize on DNA substrates. It took us, believe it or not, 10 years to work that out. Mahmood Shivji in my lab expressed and purified this entire middle section of BRCA2 containing each of the RAD51 binding motifs over 1,000 amino acids long, that fragment. And what he was able to show is that BRCA2 has opposing activities on RAD51 oligomerization on single-stranded DNA versus double-stranded DNA. In a nutshell, what we found was that in the absence of BRCA2, RAD51, when given single-strand and double-strand substrates, goes to the wrong one. It forms stable structures on double-stranded DNA, which is the wrong step. It's the second step, instead of forming stable structures on the single strand. What BRCA2 does is to stabilize the single-stranded structure, but inhibit the double-stranded structure. So if you will, it orders the reaction so it can proceed in the correct direction. We still don't understand the structural mechanism for this, but very recently, Jude Short, Mahmood, Xiaoshe Chen, and Yang Liu in my lab have been using cryo-electron microscopy to resolve now at about 3.5 to 4 angstroms the details of the RAD51 filament on single and double-stranded DNA. And suffice it for me to say, we still don't understand the structural mechanism, but we're beginning to get clues. So the point I really want to make is we now understand in some detail how loss of BRCA2 can cause genome instability and give rise to cancer by inactivating this critical pathway for double-strand DNA break repair. In fact, it's disruption of homologous recombination that also accounts for the therapeutic sensitivity of BRCA-deficient cancers to new targeted agents like PARP inhibitors or even to old drugs like platinum compounds. The repair of DNA breaks by HR is what makes normal cells resistant to the effect of these agents. So in BRCA2 deficient cells, this pathway being lost, cells are highly sensitive. So I've just realized that I've lost a number of slides, so I hope you'll bear with me. Yeah. Hundreds of variants of BRCA2 are inherited in the germline, which can give rise to cancer. And the point I want to make on this slide is simply that the majority, about 75% of these germline pathogenic alleles, work by truncating the BRCA2 protein. So how does BRCA2 truncation cause carcinogenesis? To address this issue, we decided to develop a series of genetically engineered mouse models to begin to look at tissue-specific carcinogenesis associated with these types of truncating BRCA2 mutations. We chose to do this in the pancreas, and the reason is quite simple. Unlike in the breast or in the ovary, we actually know the oncogenes that trigger oncogenesis in the pancreas. 90 to 95% of pancreatic ductal adenocarcinomas harbor an active mutant of the KRAS oncogene, so we were able to create a model in which KRAS was activated on one allele only in the pancreas, P53 was inactivated only on one allele, only in the pancreas, and we could either model the germline allele of BRCA2, a truncating allele which is found in all tissues, or specifically in the pancreas, we were able to engineer loss of heterozygosity when necessary, loss of that second allele. So if you think about it for a second, this mouse model is a little unusual in that typically people studying tumor suppressors just knock out both copies of the protein. What we have tried to do here is to recapitulate the events that occur in human carriers. A germline allele present in all tissues which truncates the protein, and loss of heterozygosity only in the target tissue, in this case, the pancreas. And what we found surprised us. What we found, and this is work going back about 8 years, done by Ferdinando Skoulidis, an MD PhD student in my lab at the time, was that loss of heterozygosity was unnecessary for carcinogenesis in this model system associated with BRCA2. Mark that. You don't need to lose the second allele. None of these mouse tumors had lost the second allele. They still got accelerated development of pancreatic ductal adenocarcinoma. It's not just a mouse artifact, because we were able to collaborate with Jon and Jorunn at the Icelandic Cancer Registry, where a large number of people in the population inherit this BRCA2 truncating founder mutation, and again we found that about 75% of pancreatic cancer arising in this population did not lose the second allele. And interestingly, but just as you'd predict, we were able to show that cancers in which this second allele persists are resistant to all the new targeted agents like the PARP inhibitors, as you'd expect, because the second copy is still functional. Recent work using genome-wide surveys of tens of thousands of cancers has confirmed our early results. This is again the WASH-U study, and what you can see here too in a study, as I said, of about 10,500 cancers, is that BRCA2 persistently violates the Knudsen two-hit paradigm. In the breast and in other tissues, between half to 60% of BRCA2 deficient cancers associated with these pathogenic germline alleles do not lose the second wild-type copy. This is a study from Kate Nathanson and Susan Domchek, and what they find again is that about half of their breast cancers in families carrying the BRCA2 mutation do not lose the second allele. So how does this work? Well, the first question we asked was, could it be that heterozygosity has some subtle, not fully penetrant effect on DNA repair by homologous recombination? In the interest of time, let me say no. These cells have no detectable defect in homologous recombination, and as I said before, they are fully resistant to PARP1 inhibitors and platinum compounds, which trigger cell death in cells lacking homologous recombination. They also lack the genomic signature typical of DNA repair defects in homologous recombination. So how does it work? Well, what I'd like to share with you is the bottom line from some studies which we concluded just a few months ago, which show, in fact, that there is a mechanism wherein we believe the environment cooperates with the genetic inheritance of truncating alleles in BRCA2, and these factors conspire to induce BRCA2 haploinsufficiency. And what I'm going to share with you very briefly for lack of time is that there's a chemical trigger that we find that leads to the induction of haploinsufficiency in cells which carry one truncated copy of BRCA2. The chemical trigger is aldehydes, and as many of you know, these are found everywhere. They're products of endogenous metabolism. They're also found, I have no doubt, in much of what we've been inhaling in our trips to and from the India Habitat Center, because they come from car exhaust fumes as well. But for lack of time, what I'd like to share with you is what we found rather than take you through all the steps in the science. What we were able to show is that aldehydes, whether synthesized endogenously or through exposure from the environment, induce a proteolytic action on a very small number of human proteins, about 50, one of which is BRCA2. This is a ubiquitin-independent but proteasome-dependent mechanism of proteolysis, and the net effect is to reduce the level of BRCA2 protein. Wild-type cells are also susceptible to this effect, but because they have more BRCA2 to start with, they're able to tolerate the effect of aldehydes quite well. BRCA2 heterozygous cells starting with half the amount of protein, although they're perfectly functional at that point, rapidly lose function. And this, if you will, induces haploinsufficiency. In other words, an environmental trigger induces haploinsufficiency in cells carrying this germline susceptibility allele, and that leads to chromosomal instability and, we think, carcinogenesis. Of course, there are many implications, not only for the potential for tissue-specific occurrence of aldehydes and tissue-specific BRCA2 deficient cancers, but also potentially opportunities for intervention through limiting or altering the way that these aldehydes are made. And I think many of you sitting in the audience will know that many metabolic processes ranging from diabetes onwards cause, if you will, changes in the way that many of these aldehydes are made in cells. So really what I'd like to convey is that diving deep into trying to understand heterozygosity and how it causes BRCA2 deficient cancers, we've found that BRCA2 can cause carcinogenesis without loss of heterozygosity, and discovered what we think is an interesting new mechanism that may underlie this. The thing to remember as well is that if so many BRCA2 deficient cancers don't lose the second allele, the PARP inhibitors are not going to be effective. And indeed, clinical experience suggests that about half of cancers are not sensitive to these types of inhibitors when they should be. And we wonder whether lack of LOH may be a contributory factor to primary resistance in this setting. So I've taken you, I'm afraid, in a rather whistle-stop fashion through some of our work on looking at germline alleles and how understanding the pathways for carcinogenesis may suggest strategies for early intervention in cancer. If I have 2 minutes, okay. I will just talk about one aspect of the work which is beginning to take place actually in India, which is why I wanted to share it with you. So many of us are beginning to understand and identify biochemical pathways that mediate disease. How does one begin to dissect these pathways to identify potential targets for intervention and to create the types of tools that can be used to test these ideas in the laboratory? So for the last 7 years or so in our lab in Cambridge, we have developed 3 groups of tools which integrate, we think, to help us to address this issue. The new methods to undertake phenotypic screening using genetic tools that essentially identify protein shapes which are involved in key phenotypic outputs. The ability to create, using structure-guided methods, chemical leads that target these types of shapes that mediate cellular functions. And finally, new kinds of microscopy that enable us to analyze the perturbation of cellular pathways after adding chemical tools of this kind. And this integrated suite of tools, we think, can help us to dissect disease mechanisms and potentially identify and validate actionable targets for intervention. This is some of the work that's actually occurring on structure-guided lead discovery at the Center for Chemical Biology and Therapeutics at INSTEM and NCBS in Bangalore. What I'd like to really end on is the answer, I hope, to what we had considered an important issue, which is cancers start from a simple scrambling of pathways to very complex traits. Can one intervene early by hitting early events to cause the loss or death of cancer cells? We think we can. And this is an example of using the types of methods that I just spoke about to develop selective chemical tools that target protein-protein interactions of a group of mitotic kinases which kills quite selectively cells which have nothing wrong with them other than that they express mutant forms of oncogenic KRAS. So for us, this has been not only a proof of principle that one can potentially intervene early in these types of settings to eliminate cells carrying oncogenic mutations, but also that the systems that we have developed to enable the discovery of these targets and the creation of chemical tools can work. So I'll stop there and say that the work that I've shared with you has largely been carried out at the MRC Cancer Unit in Cambridge, and the single slide I showed you about the work of the CCBT in Bangalore. Thank you for your attention, and I apologize for going over time.
We can have very brief 1 or 2 questions.
Yeah, just getting some water.
Hi, Ashok here. Very nice talk. I'm very glad that I was able to hear this. Followed your work over the years, and one thing that is slowly at least becoming apparent to me is BRCA2 RAT51 interaction is probably a primary event in making sure that RAT51 engages the single strand. Am I right with that? So, and because breakpoints are going to be a sort of a, will not choose where the breakpoint happens, it appears that there's no sequence context. So the question is, do you think that RAT51, this interaction along with has the DNA sequence got anything to there, or it is absolutely, doesn't care about what kind of DNA it's?
That's a good set of questions. There's more than one, so let me take them very briefly. The first is, yes, you're right. We find that in physiological ionic conditions, that without the BRCA repeats of BRCA2, RAD51 is unable to form stable complexes on single-stranded DNA. You can manipulate that a little biochemically by adding high concentrations of calcium or ammonium ions, but in physiological ionic conditions, you need BRCA2 to form that single-stranded filament. Second part of it is sequence specificity. You're right, there is no apparent sequence specificity for the repair event, but we don't know whether the break event that necessitates the repair is sequence specific. So many of the RAD51 associated lesions, BRCA2 associated lesions, occur during replication. And as you well know, there are many sites in the genome that are somewhat refractory to the transit of the replication machinery, so-called replication fragile sites, the late replicating regions, which tend to be in typically heterochromatic sites. And we wonder, but we don't know yet, whether these may be more susceptible to the breakage that then leads to mutation in the absence of BRCA2.
Hello. I have a question here. It's about aldehydes causing... Here.
I'm sorry, I'm trying to... Okay, got it.
Aldehydes causing haploid insufficiency due to a proteolytic event. So can inhaled aldehydes cause an increase in such proteolytic events, number one? And number two is, are you talking about the environmental aldehydes or like endogenous aldehydes produced during oxidative stress or something like that?
The second question is a straightforward answer, both. So what we're able to show is that exogenously added aldehydes, but also increased endogenous aldehydes, which, as you correctly pointed out, are generated during processes like, for example, lipid beta peroxidation, or even histone lysine demethylation. Many of the KDM demethylases produce formaldehyde as a byproduct. Many of these events can generate aldehydes that have an effect on BRCA2. Does that answer your first question as well? We don't know. It's certainly a possibility, which I alluded to in the context of the Delhi traffic, but we don't know for sure.
So
yes?
They say that half of our textbooks may be wrong. We just don't know which half, and I think you have shown a great illustration of a hypothesis we have held dear to heart for a long time. My question is that you have shown that the LOH is not necessary, but it still happens in half the cases. Clinically, do you see there's a difference in the patients that have LOH and others who don't?
That's an excellent question, and we don't know the answer. So basically, what we have been able to show is that the truncating mutations work by this induced haploinsufficiency pathway, we believe. What I didn't have time to share with you is that we've also found other mechanisms affecting different missense mutations in BRCA2. So we're in the process of working with collaborators across the world to identify families who carry those mutations to ask the questions which you're asking, which is, do they develop cancer earlier than the others? Is there LOH? Is there less LOH? Is there tissue selectivity? How do they progress clinically? We don't yet have the data to answer that. But let me make one point in relation to the haploinsufficiency induced by aldehydes. As I think many of you know, about 500 million people across this planet, 500 million, primarily of East Asian descent, inherit mutations in ALDH2, aldehyde dehydrogenase 2. This is the enzyme that catabolizes acetaldehyde after ethanol exposure. This is why people carrying this mutation flush, go red, they can't tolerate their drink well. It's intriguing to speculate, and we're trying to test this idea, that if they also carry BRCA2 mutations, that there may be, how do I put it, a working together, if you will, of these genetic alleles to somehow modify cancer progression, and this is something that we're testing. You're absolutely right. You're absolutely right, and I think that this is a major, major challenge for testing any potential drug-related early intervention. So the way we envision this is actually to intervene at a later stage in order to try to prove the concept. So for example, if we develop drugs which may help to eliminate RAS mutant cells, one can envision situations, for example, people who have pancreatic cancer, who have inherited mutations in the pathway, who are treated, to treat them as a secondary intervention opportunity. In other words, to see if you can delay relapse or recurrence or post-surgical grow out as a way to test in a shorter term the proof of concept for longer term intervention trials. I don't have an answer for the actual primary intervention trials. As you know, with tamoxifen, Rolodex, and the aromatase inhibitors, it's taken years and literally tens of thousands of patients to test those ideas. I think all we can do is to try to see if we can find situations in patients who have been treated and are at the risk of relapse or developing other forms of cancer, whether these types of treatments can delay or prevent progression. That's our idea anyway.
Thank you very much, Ashok. We will stop here. We will now hear about the childhood leukemia paradigm from Professor Bhaskar Saha. Professor Saha is an IndiaAlliance Margdarshi Fellow and is also the director of Tata Translational Cancer Research Center, Kolkata, and a professor of pediatric oncology at University of Manchester, UK. He leads biomarker research and clinical trials in childhood leukemia.
Can you hear me? So I think some of us worked out yesterday that this screen is useless. Okay, so I know some of you can, but the majority of people can see this screen. It's actually very difficult to manipulate both. So I'm going to do exactly what Ashok did, which is primarily use this screen. So if I wander out and block this because the projector is here, I apologize. Okay. Sorry, can we put that back on show again? Right, so the title is actually some of the objectives that were embedded in this fellowship that I began in early 2014. And that time it was at what was then a very newly opened hospital. And at the time I started, I shared the responsibility of children with cancer with one other consultant. I had no research facilities, I had no staff, and the only equipment I came with was the laptop that I had brought with me from UK. And so what I'd like to share with you today is some vignettes from the journey of the last 4.5 years. So coming back to the topic itself, what you can see here is box and whisker plots, which illustrate the differences or disparities in outcome of cancer in 3 different cancers. The red is in North America, yellow is in Asia, and I put a black bar here which shows you the differences in median. If you look down there, you can see it's differences at minimum 15 to 20% in these common cancers. And if you look at, and these are from 2 recent publications, so if you look at the differences here in India and the West, in these common cancers. With the exception of cervix, you can see the difference is actually 25%, particularly in acute lymphoblastic leukemia, where if you look at the outcomes of 90%, which is almost cure, to 65% in India. Now, one of the contributors to this disparities in outcomes in India is, of course, the cost of therapy. And centers like ours in Kolkata, through generous grants from both the government and non-government organizations, are able to heavily subsidize inpatient therapy. But patients still pay a considerable amount of money for travel and subsistence. And if you look at over here, this white bar over here is the outpatient costs. This is the scale 150 to 200% of patients coming from rural areas who are coming to hospital. And this is so the cost of out-of-pocket costs can be substantially more than the cost of therapy. The further the patient travels, the more the expenses go, and this for a low middle income family can push them into poverty for a generation. So that's a different problem. Now coming back to acute lymphoblastic leukemia, so this is the outcomes of 3 different Western and 1 Asian study group in this decade. This is the survival over here, and you can see the survivals are all in the mid-80s. And this is actually the results of incremental improvements in outcome using the same drugs for the last 5 decades by first intensifying combination chemotherapy in all patients. Problem lights? Too bright, no? Yeah, if you can bring the brights on, yeah, if possible. So if what we subsequently discovered is that there are groups of patients identified by clinical criteria or cytogenetics who are at higher risk for relapse, intensify therapy for them further. And in this modern era, we now have molecular tools that can detect residual disease after treatment. And in patients who have less than optimal response to therapy, we intensify therapy further. Okay, so this risk stratified therapy. So how have we done so far in India? So this is the outcomes of patients in India. You can see the outcomes here at the bottom, which is about 60% to 65%, which is 20 to 25% different over here. And actually, this is the earlier results. So results have not really improved in the last 3 decades. And if you look at the reasons for this, so this is PGI Chandigarh has decreased the relapse rate from 27 to 15%, but overall this treatment-related mortality still remains the same. So why are patients dying? So there are some reasons for this. One is all these groups use risk stratified therapy using the criteria we showed you here, and this risk stratification limits or decreases the intensity of therapy for patients at low risk of relapse. Centers here in India have not been using this kind of therapy, and they are then forced to treat all patients with moderate to high intensity therapy, which relates to this treatment deaths. Furthermore, if you look at these groups, these are all multi-center studies which have large cohorts, okay, which allows adaptation to events within the trial itself. Single centers which have smaller cohorts cannot adapt to this, okay, and they will only be wiser after the event. But there's one more factor that I would like to bring out to you, and that is that if you look at this, you can see there's a long recruitment time, 7 to 10 years, and the gestation times to publication. So the average time from recruitment to publication is 10 to 15 years before the data actually comes out, okay. Many things happen during that time. Many changes happen in the clinical trial. And that's because these groups all work together. Not only these groups work together internally, they also work across. So the Germans know what the Americans are doing, and the British know what everybody else is doing. So at the end, when the publication actually comes out, it only reflects a small piece of information. So what we really need here is open access to ongoing studies. Open access to publications does not help. Okay? So what we did to try to address some of these problems is we brought together all the major pediatric oncology centers in the country, and they're here. This is where all the patients go to. And prior to the study of the fellowship, start of the fellowship, we organized a common protocol and agreed to this. We played to the strengths of the centers by helping each center, the one center as a nodal center to standardize the MRD, cytogenetics, and the clinical trials. And we also were very lucky to get the generous offer from the international group, where waived the cost of registration and participation in closed workshops, but our participants still had to pay the cost of travel, which they had to pay from their own pockets. But nevertheless, what happened at least was equity. Patients no longer a lottery of where they were going. All centers treated uniformly. It also allowed patients to opt to stay at their local center, but more importantly for patients who actually went to a different center, say from Kolkata to Bombay, they could come back at any stage of their treatment and just carry on from where they had left off to. So just to show you what the strategy is then, we divide the patients to standard, intermediate, and high-risk group based on cytogenetics and the clinical characteristics. You can see for the first 5 weeks, the low-risk patients get less intensive treatment, the high-risk get more intensive treatment. At the end of 5 weeks, we do a bone marrow and we measure the minimal residual disease. The children who have lower disease level less than 1 in 10,000 carry on with less intensive therapy. If they have higher disease than 1 in 10,000, they join the high-risk group. And that decreases the treatment intensity for this group of patients. But in spite of all this, this is the first sort of 6 months of treatment. You can see that the majority or the longest duration of treatment is this 2-year treatment of maintenance. So what happens during maintenance? So during maintenance, all children receive daily dose of 6-mercaptopurine. The thiopurines have a wide pharmacokinetic variation, and this requires constant adjustment of dose. And we do this by monitoring the side effect of the drug, and that is neutropenia. We try to maintain the child's neutrophil count between 1,500 and 750, and this is done by fortnightly blood counts, sometimes weekly. And to avail of this, there are families who will stay next to the local hospital for 2 years, okay? And I realized that even families who are just coming to my clinic, that majority of my families were spending an overnight trip coming, an overnight trip going for a 10-minute visit to the outpatient clinic, okay? So what we began to do is we began these patient health records first, which actually they put the count in, they put the dose in, they scan it. There are kiosks everywhere, even in small rural areas. They can email it to us, and we email back the dose to them, okay? And we monitor this, and that way actually we're capturing the data as well. We're now beginning to develop this as an app that they can do and use. But one of the things we realized is that clinicians who are doing this are very quick in decreasing the dose or stopping the dose in somebody whose counts are dropping. But they are not escalating the dose in children who are not getting the optimal dose. So we decided to look at this. And the way we did this was we took 46 patients in my clinic who had completed 2 years of therapy. And this shows you each dot is the average mean weighted neutrophil count for the entire 2-year period. So it's about 6,500 observations in all, okay? What you can see over here, this is the target. The target neutrophil count should be between these 2 lines. And you can see some of them, but not all. And this is the mean weighted dose. And what you can't see here because the light is too bright is that there's a yellow box here, and actually this is where most of the patients should lie, but they're not. So we were very good at stopping treatment in patients whose counts are, but we were not escalating therapy. And that can actually change outcomes by 10% if you don't optimize the therapy. So we've now got a system by which these counts are automated in the clinic because the clinic is very busy, and this comes into an app which is on the doctor's phone, and this tells a doctor how much to prescribe, okay, based on the data that they've got. Because the tablets are 50 and 25, and that's all we can get. This probably produces a printout for the patient, so the patient has a visual, prescription to see what is happening. And for patients who actually have an Android phone, which is a smartphone, we can put this app onto them, which gives you a daily alarm saying this is how much you need to take, this is what you take. And when they press the button, we get a record that they've actually taken their medications, and that's another study. Now, how have we done so far with this study? So this shows you the treatment-related mortality historically. We ran a 3.5-year pre-trial study and now a randomized study. And you can see as the groups have got more familiar with the protocol, mortality is now as per sort of close to Western standards. Relapse rates have decreased as well. This 1% is too early because there's only first-year data. Outcome data then, so this is the whole cohort now. This is 3-year study, 3-year survival. We need another couple of years before we know where this stabilizes. But still, this is much higher than has been achieved before. And the whole focus was in getting the best outcomes in the standard risk by delivering least amount of therapy. There was a lot of nervousness among the Indian investigators, worrying that there will be a high relapse rate. But we are approaching 90% or over 90% outcomes in the standard risk group by giving them the least intensive therapy. So far, so good. But we have a couple of problems. One is if you look at this cytogenetic subgroup, this intermediate subgroup, which is actually a diagnosis of exclusion, what you find is this about half the population, which shouldn't be more than 40%. So we're seeing more intermediate risk groups, but this also may be because in the earlier days of the study, cytogenetics was not standardized and the centers were not able to identify the good risk patients. But if so, then we shouldn't see this 15% difference in outcomes between the good risk and high risk. It should be higher. So either we are seeing more, or we have a more high-risk category in this intermediate risk group. And the second was in this MRD subgroup. So high MRD should be seen in 40%. We're seeing it only in 20 to 25%, which suggests that the tests that we have standardized are actually not as sensitive as we thought they were. So we began to start looking at this more closely in the Kolkata cohort in my center, because we were been investigating them more closely. So if you look at the distribution, cytogenetic distribution, this is the UK distribution. This blue and the yellow are the high hyperdiploid and ETV6 rungs 1. These are the patients who have 90% survival, and these are low risk. And you can see clearly that our cohort is smaller, particularly in the ETV6 rungs 1, probably not as smaller as was indicated by the cohort study, but high risk is about the same. The intermediate risk group is a little bit bigger. But what stands out here, if you notice, is that we see a larger proportion of patients with high hyperdiploid compared to the West. Now, high hyperdiploid by itself is a heterogeneous condition. All it says is that it's a condition where the nodal number of chromosomes, there's a whole gains in chromosomes in era mitosis, which is 51 to 67 chromosomes, and these are the commonest chromosomes that are duplicated. So we looked at this with a high-density SNP array. So the patients here are in rows, and the copy numbers are in columns. I'm afraid the light is not allowing to see them very clearly, but what you can see here marked in the asterisks is that we see the same chromosomal duplications. This is also showing you a different kind of loss of heterozygosity from what Ashok showed you, which is how the chromosomes are duplicated, uniparental isodisomy. So the chromosomes that are coming on are coming from one parent, and that's being duplicated, and the other parent is lost. Okay. Now, in the West, what has been reported is trisomy 11 occurs in 10%. We see this in 25%. But the most striking feature is that in the West, what has been reported is that whole chromosomal uniparental isodisomy is seen in non-random fashion in 8%, and in chromosome 9, 7, 11, and 12. We are seeing it in 48%, and it's a very random pattern. Okay, we can't really say which chromosome. So the pattern is actually quite different. Okay, if you see, look at chromosome 18. Okay, we see almost all our patients carry duplications of chromosome 18. This is associated with EFS of 90% in Western studies, but actually a third of our relapses in our cohort are coming from this high hyperdiploid type. So all indications is that, at least at the epigenetic level, we're seeing a different high hyperdiploid cohort. I mean, certainly in the treatment response pattern, we are seeing a different high hyperdiploid cohort. So we've been also mapping this at the somatic mutation level. And again, you can't really see this, but these are the genes which are mutated in our targeted exomic sequencing profile. In red are all the genes that have been identified in this group of high hyperdiploids. So we see them all. But the frequency of genes that have not been detected, and these are much higher in our cohort, which does suggest that at least at the gene expression level, we are going to see a different expression in these high hyperdiploid cohort. Gene expression will correlate to treatment outcomes, and that's what we're currently investigating. So moving on from that was the question of MRD, right? So the flow cytometry-based MRD is used throughout the centers because it's cheap, it's rapid, everybody's got a flow cytometer. We set up a genomic-based PCR, which is used as a research tool, but we also use it diagnostically for relapsed patients, because relapsed patients who have high MRD after the first block of therapy become eligible for transplant, and so therefore an expensive test is worth the cost. So I just want to show you this one. This is the end MRD levels at the end of first block of therapy. This is flow cytometry. Blue is the ones who are below 10 to the power minus 4. So in this, you would say that 80% of patients are not eligible for transplant and may be cured by chemotherapy. If you use the genomic PCR, this drops to 40%. Okay? So clearly, these patients are a much higher risk of relapse, and are not being picked up by flow cytometry. If you look over here now, PCR is actually being replaced by high throughput sequencing, and this red cohort is a group of patients retrospectively analyzed by the American Children's Oncology Group using an Illumina platform, where they identified patients who are positive by high throughput sequencing but negative by flow cytometry, who have a different outcome to compare to patients who are negative by both techniques or positive by both techniques. Now, the Illumina platform is a little more complex and expensive, and we've started exploring about whether we can use this nanopore technology to actually do this, because one of the big problems we do have is that this technology of MRD and cytogenetics is not available at most centers. Flow cytometry uses fresh cells, and this is difficult to transport. A genomic solution to MRD and genetics will allow us to risk stratify patients across the country, across all centers. Now, the question about MRD that is there is that why do you have a small proportion of cells that are still surviving? And the one obvious suggestion to this is that you have subclones which have different genetics, okay, which have subclonal mutations acquired after the first initiation event, okay. Now, the most common subclonal events that you see are deletions or mutations of the developmental genes, cell cycle genes, or signaling genes, and they're listed here. I'd like to just draw your attention to this one, which is IKZF1 and deletions. And that's the one that has been prognostic across many different series. Not, doesn't matter how you treat them, different groups across the world have reported this is prognostic. And you can see this in the group recent report by the Italian and German group. And this is a group of patients who are defined as Icarus plus. So they have an Icarus deletion plus a deletion of any of these other genes. And these are the ones who have the highest relapse rates. But even there, you can see that there are patients here who are MRD negative, and they actually have decent outcomes. So there are some other factors that are going on here. So you investigated this now in a relapse cohort, because that's where you expect these clones to now get enriched, and you should be able to now pinpoint now which are the cells, which are the mutations and cytogenetic changes that are coming out. So you can see this is low-risk cytogenetics, intermediate and high. This is, the green is low MRD, high MRD. And as you'd expect that from going from low to high, the proportion of MRD is increasing. So this is 50%, it's about 65%, and here's almost 80% of patients are high MRD. If you look at the subgenetic changes, which is the Icarus, PAX5, etc., what you can see here is that in the MRD positive cohort is now getting enriched with deletions of Icarus, PAX5, NR3C1, CDKN2AB, and mutations of NRAS, and not the others. Now, what would be the potential, relevance of that, and I'll come to that. The other thing we did also was to look at the outcome of these relapse patients as they went through, and saw what emerged in the second relapse. And you can see that MRD predicts what happens to these patients very clearly. But surprisingly, none of the other mutations came out. The only thing that came out was mutations in P53. Okay, so patients who had mutations in P53 were the ones who did poorly, and this is either mutations or deletions. And actually, P53 mutations we hardly pick up at all at diagnosis. It's only coming at relapses, less than 2% and 13 to 15% at relapse. So that suggests there's an enrichment of a low-level P53 clone that is actually coming out, and that is drug-resistant. Now, to put all this together then, what is the commonality between all these genes? So there's a recent paper showing, and this is some of the work we've also been doing, that leukemia induces a glucose resistant or insulin resistant state, okay, which allows a lot more of glucose to be available to leukemic cells. And actually, even though these are transcription factors, the way they regulate this Icarus 5, NR3C1, PAX5 work together to actually downregulate glucose metabolism in a cell. This allows the B cell to differentiate and stop proliferating. If these are deleted, the B cell continues to proliferate, doesn't differentiate, and enters a state of drug resistance. Okay, so this is a paper by a group in California. And if you have deletions in this cell cycle genes as well, you get proliferation. And work done in our lab and other people's labs also suggests that P53, regulates the cell cycle, but also regulates glucose metabolism, and probably in very similar ways that this does, which suggests that actually targeting metabolism rather than targeting mutations might, so in many ways, in a parallel to Ashok's story as well about the aldehyde. So coming back to where we are now about equity. So we said that we've actually shown that we can deliver cheap, low-risk therapy to a large group of patients using modern technology, but it's actually only affecting 10% of patients. The majority of centers are using our protocols but cannot do the risk stratification. But we've shown also that genomic-based technology can actually do all of this and actually can continue to refine this. So the idea we've got is that if you have centralized laboratories where you send your genome, or a piece of DNA, rather than the patient, you can actually make a diagnosis, you can stratify the patients, you can keep the patients where they are. We've developed clinical trial management systems that are actually helping the centers decide how to treat their patients, and including patients taking charge. So the patient stays there. And through this process, you can identify high-risk patients who actually need to travel or only need to come for blocks of therapy. Okay? And that's the proposal we have for acute lymphoblastic leukemia, which we think would probably work for any cancer in this country. So what we've shown you so far is that the approach that we took up, that if you work together, you develop network, and you brought in technology, you could actually make things cheaper for patients, because one of the arguments always is technology is expensive, but actually the cost of the technology is trivial compared to the cost of therapy. If you develop a network system like this, we can find solutions that can be used for all patients. But more importantly, I think from the laboratory, we're now finding that there are possible alternative methods by which you can target these leukemic cells, either to make cheaper therapy by repositioning or repurposing drugs that are already existing, metformin, for example, which might work in glucose therapy. And that can offer much better options to our patients where cost of therapy is escalating rapidly. And so finally, this is where we are now. So this is where the hospital was when we started. This is a new wing that is opened. The pediatric patients have one entire floor to themselves. We have now 6 consultants instead of 2. 50 beds, one of the largest units in the country. And behind that, now we have dedicated research facilities. And now to get to this journey that we have come through hasn't been an easy one. And I have to thank many people, more than fit on the slide, both at my center, in India, and elsewhere, but primarily from people from outside, my colleagues from abroad, many of them who have come and spent time at their own costs, paid their own fares to come and teach and train and help us through this process and continue to do so. But I'd also like to thank Tata Consultancy Services, who not only helped us get this laboratory started, but have been integral to our development of the knowledge pipelines, both for the clinical side and for the genomic sides. And I'd like to thank IndiaAlliance, not just for the funding, but also for the fact that the flexibility with the funding. But more importantly, it's been such a great pleasure. From the start of the application, under the lawyer, clear dictate as what was expected from the IndiaAlliance led to a successful application. The current CEO has always been extremely responsive to any queries. I can send a query at 8 o'clock at night and I get a response back. And to the nodal officers who have always worked out saying that, okay, we got a problem, but you leave it with us, we'll find a solution. Okay? For 25 years or more than that, a quarter century of working with different grant committees, this is the best one I've ever worked with. Okay? So what I would like to say today is an old Indian phrase. Okay? 10th birthday, 100 saal jiyo, which is, may you live for 100 years. Okay? And I'll tell you why. We researchers in India need you. Okay? More than you actually think you realize. No, okay? So all the best, and thank you very much. I haven't woken up for breakfast yet.
Interesting talk. Yeah. Hi.
Hi.
Interesting talk. I just have I treaded a little bit away from what you were talking about, and I realized that drug resistance seems to be a problem even with cancer therapy. So when you consider relapse, is that in any way, have you considered relating it to development of drug resistance in this, that being a cause for relapse?
Yeah, it's probably too big a topic to talk about. Yeah, it's drug resistance. So basically you define drug resistance very simply that you target a cell and the cell survives, right? Okay. Now the question is why the drug resistance, and I think Ashok alluded to that in I think his first or second slide, which he said it's the genetic constitution of the person, the genetic constitution of the tumor, and the environment. Okay, and certainly I think that's common to all mechanisms of drug resistance.
So you've covered like the deletions and the further mutations that are happening. All that has been covered in that umbrella, basically.
I haven't talked about our work with the microenvironment and how we are dealing with how the microenvironment deals with drug. We have published extensively on that, but 20 minutes is too short a time to fit that into the story. So.
Thank you.
Hello. You mentioned, it's me. You mentioned something very important, that it's not enough having open access to the publications. You need open access to studies. And I think that applies for science in general. And I have two questions. Number one, isn't that achieved, at least in clinical trials, through clinicaltrial.gov registration? That's generally mandated for clinical trials. And secondly, what are the statistical implications when you release a study too early? There's a quality issue. You are affecting the power of future studies. So how should all that be taken into account?
Yeah, I think the simplest explanation to your first question is that no, because you have to be there in the room when people talk about unpublished data and work with each other saying, what does this data mean? So it's a bit like, you read Ashok's excellent paper. None of us are going to be able to do this work by reading his paper without even done any laboratory work at all, okay? So it's not, you have to be there. You have to learn how negative decisions were taken. We're not going down this line. That decision actually might work for you, but not for Germany. And you have to be there in that room. And the second question that you had was about, sorry, the statistical. Yeah, so statistical implication is we're moving away, and actually, this is my belief, we're moving away from these large-scale randomized control trials, okay, into the N-of-1 kind of basket kind of things that are happening in the West. This is much more difficult over here. And in terms, again, by being within the groups that have statisticians that have been working with these problems for decades, okay, I've been struggling here. My statistical support still comes from UK because I've been struggling here to find a statistician who understands the kind of clinical trials we run and what are the issues over there. And Gautam's have been nodding his head because I've been talking to him about it as well. But I think that is surmountable, but the first one is not. No, that's not true. That is not true at all.
But because that's where the problem is. Because in a sense, your power is based on the final pre-specified analysis.
Correct.
So when you do more frequent analysis, you're reducing the power and you're increasing the, let me finish. Okay.
Yeah.
You're increasing the chances of false positives. Nothing wrong with it, but that needs to be built into the statistical thinking. And this may be where we have to develop an entire body of Bayesian statistics for dealing with trials.
Yeah, you're preaching to the converted about Bayesian statistics, but we build into because if you look at how we are required to report trials now, we have to actually give the pre-analytical statistical study that's there. In all of this, there's always a question, will you recruit enough patients to the study? So we have interim analysis built into that, which we have to report back to the data monitoring committee, which will say the study is futile, you're never going to recruit, so we close it, okay? So those decisions are built in. As you go into these sub-variables of what happens to the mutations, then you have to use probability and Bayesian studies to start dissecting out what is happening. And as you said correctly, they have to be built in right from the beginning and not done post-hoc.
One last question from Srilaja.
About polyploidy.
I think I missed it when you said it. So in your hyperdiploid analysis, the loci that turned hyperdiploid included the causative loci for those kinds of leukemia?
As far as we know, the high hyperdiploid is an initiating event because you can pick this up in Guthrie cards from cord blood in patients who have not yet. In such a study is actually, I don't think
it's hard to
look at exactly all the events that have happened, though I guess modern technology might be able to do that, but we've not looked at that.
Because with chromosome painting, etc., maybe you could tell exactly which ones have turned hyperdiploid.
The high throughput SNP would tell you whether it's the same thing that you're looking at further downstream. It's a very interesting question. I'm a clinician. I don't really care how it works, how it happens. I want to know if I can shoot it down.
Right. Thank you very much. We will move on to the next speaker. Our next speaker is Dr. Kavita Babu. She is again our IndiaAlliance Fellow. She is an intermediate fellow and an assistant professor at ISER Mohali. She is a neurobiologist who works on molecules that regulate synaptic functions. She will talk about how a neuron talks to its target.
Okay, I think you can hear me. First, I'd like to thank the Alliance for giving me the opportunity to talk about some of our work, and of course for the money. And I'm going to tell you a little bit about what what we work on in the lab, which is essentially to look at the molecules and the mechanisms that are required for a neuron to talk to its target. And I'm going to be talking about the target in this case is going to be the muscle. So we're talking about neuron talking to the muscle at the neuromuscular junction. So as all of you know, the brain is required for us to do everything, be it thinking, walking, talking, waving, everything requires our brain. And the brain is made up of billions of neurons, which form many, many connections to other neurons, giving rise to trillions of these connections, which are called synapses. So how does a neuron talk to another neuron or its target? You essentially have, oh God, this does not work. You essentially have an electrical, sorry. You have an electrical signal that comes along the axon. This causes calcium influx at the presynaptic site, and this then causes release of a chemical which is called a neurotransmitter. The neurotransmitter binds to the postsynaptic receptor, which is present in the postsynaptic cell, which is the cell this neuron is communicating to. That then activates the postsynaptic cell and causes changes. If it was a muscle, it could cause the muscle to contract, or relax. If it was another neuron, it could tell this neuron to then cause another neuron to show changes in activity. So as I said earlier, our brain has billions of neurons, around 10 billion neurons. It's extremely difficult to think about studying 10 billion neurons, and especially at the level of molecules, to actually figure out who the molecules are that allow you to make, that are required for synapses to function properly. So in order to simplify the system, we use this elegant organism, which is called Cinorhabdatus elegans, or C. elegans for short. And this has 302 neurons, making it fairly straightforward to start looking at which neuron talks to the other neuron and the molecules involved in this process. There are more than 10 trillion synapses in the human brain, while there are around 7,000 fairly stereotypical synapses in C. elegans. However, although there are very few neurons in synapses, the number of genes that encode for proteins in C. elegans is very similar to the numbers that are present in the human body. And a lot of the genes that are required for making different neurotransmitters, the neurotransmitter receptor proteins, the auxiliary molecules, cell surface molecules that are required to bring the synapse to its target, all of them are conserved in C. elegans, making it a very nice system to start asking questions as to who are the molecules that are involved in different processes. So I'm going to be talking today about two very short stories in our lab, and both these stories deal with how the neuron talks to the muscle. I'm going to talk about how we found two molecules which are involved in this process, and before I begin talking about the stories, I want to give a brief introduction to an assay that we use, which is a very simple assay that allows us to look for defects in neuromuscular junction functioning. So, as I said a little earlier, you have a neuron that synapses onto the muscle. This neuron in C. elegans sends out the neurotransmitter, which is called acetylcholine. Acetylcholine binds to an acetylcholine receptor, which is present in the muscle. This then allows the muscle to contract. And this is one of the mechanisms by which the worm can actually move on any surface. So apart from acetylcholine binding to the acetylcholine receptor, you also have an enzyme called acetylcholine esterase. This enzyme breaks down the acetylcholine to prevent too much acetylcholine from accumulating at the synapse. Now, if one were to break down the acetylcholine esterase using a drug which is called aldicarb, you can imagine that you'll have a lot more acetylcholine at the synapse. This would then cause too much activation of the muscle, or too much activity in the muscle. The muscle would keep contracting. And the end point of this is that the worm paralyzes. And with specific concentrations of aldicarb, you get very nice curves of paralysis, where you have no paralysis at the 0-minute time point. Around 60%, sorry, around 25% of the worms paralyze at the 60-minute time point, and all the worms are paralyzed by 120 minutes. So you can imagine that you can use this assay to knock down genes and look for mutants that cause either increased paralysis or decreased paralysis. So one of the set of molecules that we're interested in looking at were claudins. A lot of work has been done in claudins at the epithelial cells. Claudins are required to bring two epithelial cells together. Essentially, claudins are 4 transmembrane domain proteins which have 2 extracellular domains, and they bind to another claudin which is in the neighboring epithelial cell, bringing these 2 epithelial cells together, forming a junction of sorts between the 2 epithelial cells. However, what is known about claudins is that they're also required in the blood-brain barrier. A lot of work is now being done to inhibit claudins and allow for molecules to easily access the blood-brain barrier. Apart from this, mutations in claudins have also been found in Alzheimer's and different neuropsychiatric diseases. And claudins are also expressed in neurons. So this indicates that claudins might have functions other than what we already know in the epithelial cells. They might be involved in aspects of neuronal function and synaptic function. So we wanted to find out if claudins, at least in the case of this organism in C. elegans, are required for synaptic function. So what we did was we looked at all the claudins in C. elegans. Humans have 24 claudin genes. C. elegans has 18. So we knocked down each of these 18 claudin genes in C. elegans. And we went on to see whether any of them caused increased paralysis on aldecarb or decreased paralysis on aldecarb. And what we found was that a couple of the genes that we knocked down showed increased paralysis in aldecarb. This is the wild-type paralysis curve. 2 of them showed increased paralysis, and 2 of them showed decreased paralysis. All of the others were normal. So we then decided to work on these molecules, and I'm going to tell you 2 stories. One on this molecule called HPO30 that doesn't show any paralysis on aldecarb, and the other, which is called HIC1, that shows increased paralysis on aldecarb. So once we started working on HPO30, we first thought, what could be the reason for decreased paralysis on aldecarb? So when you think about it fairly simplistically, there are 2 obvious reasons. You have less acetylcholine release, that could cause the worm to paralyze slower on aldecarb. And the second would be less postsynaptic receptor, which would also cause the worm to paralyze slower on aldecarb. So we thought, okay, let's first see where HPO30 is required for this function of paralysis on aldecarb. And when we did our initial experiment, we put back HPO30 in neurons and in the body wall muscle. And what we found was that HPO30, when we put it back into the body wall muscle, could completely rescue the phenotype seen in the mutants, but not when we put it back in neurons, indicating that HPO30 functions in the neurons. We then did experiments to look at the HPO30 expression and found that it was indeed expressed in the body wall muscle. So then we decided to ask, okay, who is HPO30 affecting? Is it affecting the acetylcholine receptor that's present on the body wall muscle? In order to do this experiment, I just want to briefly take you through the 2 different types of acetylcholine receptors that are present on the body wall muscle of C. elegans. So one of them is the alpha 7 type acetylcholine receptor, which is a homomer of 5 different subunits, which is called ACR16. This is 50% homologous to what is present in humans. The other is a more specialized type of acetylcholine receptor, which is essentially present in nematodes. It's a levamisole-sensitive acetylcholine receptor, and it is made up of 5 different subunits that come together to form a heteropentamer. So we looked at the different acetylcholine receptors, and we found that the heteropentamer, or the levamisole-sensitive acetylcholine receptor, was the one that showed decreased expression in the HPO30 mutants. We looked at the different subunits. All of them showed decreased levels at the synapse in the HPO30 mutants. And again, when we put back HPO30 into the body wall muscle, we could completely rescue this phenotype. So we then asked, okay, if HPO30 is indeed affecting the levamisole-sensitive receptor, one would then imagine that these mutants would be resistant to levamisole, which is another drug which is used as a nematocide. So when we looked at HPO30 mutants, we found that they were indeed resistant to levamisole, and you put back HPO30 in the body wall muscle, you can completely rescue this phenotype. So all of this indicated that HPO30 is affecting these levamisole-sensitive receptors. We wanted to see at what point it was affecting these receptors. We looked and did experiments to look at whether physiologically the activity of the receptors were different in these in HPO30 mutants. And these experiments were done by filleting open the worm and patching onto the muscle to form a patch clamp or a whole muscle patch clamp, and then puffing on levamisole onto the body wall muscle and looking at the current. You can see that in wild type, you get an immediate current on puffing on levamisole, which is the levamisole current. And in HPO30 mutant, this current is almost completely lost. Again, we put back HPO30 in the body wall muscle, we can completely rescue this phenotype. So all of this essentially indicated that HPO30 is functioning to somehow affect the acetylcholine receptors at the body wall muscle. It did not seem to be affecting either the levels of the transcription of these receptors, as we saw by quantitative PCR experiments, or the amount of receptors which we detected using antibodies against 2 different subunits of these receptors. So then we thought, okay, what could HPO30 be doing? We did a number of co-localization experiments and co-immunoprecipitation experiments, and what we found was that HPO30 is essentially forming a complex with these receptors and allowing for the stability of these receptors. So essentially what happens is that HPO30 sits in the body wall muscle along with these levamisole-sensitive receptors, and when HPO30 is not there, these receptors kind of drop off the body wall muscle and lose their integrity. So this then gives a large number of unanswered questions as to what is the mechanism, how is HPO30 doing it, and what are the other molecules that are involved in this process. And we are in the process of studying some of this. The second molecule I wanted to talk about was a molecule that we have very unimaginatively named HIC1 for hypersensitive to inhibitor of cholinesterase, which was essentially a molecule that we found in the screen that showed that paralyzed faster on the drug aldicarb. So we started working on this molecule, and the first concern that I had was that this molecule essentially made the worm paralyze faster on aldicarb. One can imagine that the epithelial cells are somehow disrupted in these mutants, and then that then allowed more aldicarb to get into the worm, and that would then allow the worm to paralyze faster. So in order to actually look at that, what we did was we looked at dye uptake assay. So essentially you put the worms on dye, look at how much of the dye gets in. Usually if the epithelial cell is normal, you don't see any defects in the dye uptake. And what we found in these mutants was there was no defects in the dye uptake. I'm not showing that data here. So the next question we asked was, if indeed HIC1 was affecting either the neurons or the muscle, could we rescue HIC1 by putting HIC1 back into neurons or muscle? So we did an experiment where we put HIC1 back into neurons, muscle, and into epithelial cells. And what we found was that when we put HIC1 back into neurons, we could completely rescue the hyperparalysis phenotype seen in the mutants. When we put HIC1 back into muscle or epithelial cells, we could not rescue this phenotype. Again, we put HIC1 back very specifically into acetylcholine neurons that synapse onto the muscle and GABA neurons that also synapse onto muscle. And we found that putting HIC1 back into acetylcholine neurons completely rescued the phenotype, but not putting HIC1 back into GABA neurons. So this then indicated to us that maybe HIC1 is functioning presynaptically in muscles to affect synaptic function. So then we asked, okay, where is HIC1 localized? We did a whole slew of experiments and found that HIC1 is essentially localized in acetylcholine neurons as well as in acetylcholine synapses. And here you can see co-localization of HIC1 along with a synaptic marker, synaptobrevin, which is specifically expressed only in acetylcholine neurons and synapses, and you can see that they co-localize very nicely. So we thought, okay, HIC1 is expressed in acetylcholine synapses and seems to be affecting the neuromuscular junction in acetylcholine neurons. So what then would be the cause of hyperparalysis on aldicarb? Again, when you think about it fairly simplistically, one reason could be increased acetylcholine release, and the second reason could be increased acetylcholine receptor levels. And well, we did a large number of experiments, and what we found was that the acetylcholine receptor, but in this case, the homomeric acetylcholine receptor, which is called ACR16, which is a conserved receptor from worms to humans, is what is increased in these mutants. So HIC1 mutants show increase in the acetylcholine receptor, ACR16, and again, putting HIC1 back into acetylcholine neurons could completely rescue this phenotype. So the question we now asked was, why is it that we can put HIC1 back into neurons and completely rescue a receptor phenotype? So far, I've showed you that essentially HIC1 sits presynaptically at acetylcholine synapses, and in the absence of HIC1, you get too much acetylcholine receptor. So we thought, okay, why would HIC1 be sitting presynaptically and affecting receptor levels? So we went back to look at what were the differences and similarities between the levamisole-sensitive receptor, which I showed you earlier, and the acetylcholine receptor ACR16. And we found that essentially there is nothing in common with how these receptors are trafficked, with how these the biogenesis of these receptors, and how these receptors are maintained at the body wall muscle. So what causes changes in the ACR16 receptor was completely different. It was all presynaptic changes that caused changes in the ACR16 receptor, and only postsynaptic changes show changes in the levamisole-sensitive receptor. So we thought, okay, let's see what happens in the case of acetylcholine receptor ACR16. What we found was that a beautiful paper from Villou-Marique's lab showed that Wnt signaling Wnt is a small molecule that has been shown in many different developmental and neurobiological processes. And essentially, Wnt, which is sent from the presynaptic neuron, binds to its receptor, which is present on the body wall muscle. And this then goes through a pathway involving disheveled and allows for acetylcholine receptors to get onto the surface. What is downstream is still very, very unclear. Previous work during my postdoc and from my lab at IISER Mohali has also shown a very similar situation where you have Wnt release binding to a non-canonical receptor as well as the canonical receptor and allowing for acetylcholine receptor to get onto the surface. So the question we decided to ask was, does HIC1 then function through the Wnt signaling pathway? And to do this very simply, all we did was remove HIC1 and remove different members of the Wnt signaling pathway, including the molecule that's required for release of Wnt, which is called Wintless, and the Wnt receptor frizzled. So if the Wnt signaling pathway is required to bring acetylcholine receptor onto the surface, you remove the Wnt signaling pathway, you would get less acetylcholine receptor on the surface. The worms would then paralyze slower on aldicarb. And that was indeed what we found. When we removed frizzle, the worms paralyzed slower on aldicarb compared to wild-type worms. And when we removed HIC1 in the presence of frizzle, we saw no difference between frizzle mutant and the HIC1 frizzle double mutant, indicating that HIC1 could be functioning through the Wnt signaling pathway. We looked at a whole bunch of other molecules, and we saw exactly the same data, that HIC1 was going through the Wnt signaling pathway. So the next question we thought of asking was, okay, if HIC1 is going through the Wnt signaling pathway, how is it doing it? And one obvious way it could be doing this is HIC1 could be affecting Wnt release. In order to look for Wnt release, we used a method that was previously used by Villou-Marique's lab in 2012, where they tagged Wnt with M-cherry, which is a fluorescence marker, expressed it in cholinergic neurons, and then looked at these phagocytic bodies, which are called slomocytes. So any small molecule which is sent into the pseudocilum of worm is taken up by these phagocytic bodies called slomocytes. So you can essentially look at fluorescence within the slomocytes to see how much of this molecule is secreted. So we did this with HIC1 mutant. I'm sorry for it's not very clear here. We saw that in HIC1 mutants, there was a lot more Wnt that seemed to be secreted compared to wild-type animals. And when we put back HIC1 into acetylcholine neurons, we could completely rescue this phenotype. So we also looked at another Wnt, and we see the same phenotype, indicating that this is a process which is fairly conserved, where any Wnt that's sitting in cholinergic neurons, in the loss of HIC1, shows more release. So we then asked, if this is indeed Wnt, if we were to remove the molecule that's required for release of Wnt, can we completely suppress this phenotype? And the answer is yes. If you remove use the Wnt-less mutant, you see almost no release of Wnt, and you put Wnt-less along with HIC1, again you see almost no release of Wnt. So all of this indicates that a claudin is sitting presynaptically at the neuromuscular junction and causing Wnt to get released. So the next question we wanted to ask was, how could a claudin-like molecule sit presynaptically and affect Wnt release? And what we found, we went through literature and we found that claudin molecules bind to actin binding proteins called zona oculans and affect the actin cytoskeleton. And that is exactly what we found. We found that HIC1 mutants show a decrease in the stability of the actin cytoskeleton using actin binding protein markers, as you can see here. And again, we could rescue this phenotype by putting HIC1 back presynaptically. So it looks like HIC1 somehow functions to affect Wnt release by affecting the actin cytoskeleton. The next question was, if we were to break down the actin cytoskeleton, would we see increased Wnt secretion? And the answer is yes. If we break down this as actin cytoskeleton using the drug letrunculin A, we see increased Wnt secretion. So what is known about Wnt and the actin cytoskeleton? Almost nothing is known in literature. However, there was a very nice review from Vivian Budnick that indicated that the actin vesicles that contain Wnt are stabilized by the actin cytoskeleton. So there's a possibility that breaking down this actin cytoskeleton causes the Wnt to get released more. So the next question we asked was, who is the molecule that HIC1 is binding? Because HIC1 itself does not bind actin. We went through a huge process where we looked at all the different molecules in C. elegans that might be affecting, that might be functioning between HIC1 and the actin cytoskeleton. And we narrowed it down to a single molecule called neuribin-1. The mutants in neuribin showed exactly the same phenotype as HIC1 mutants. And using a split GFP analysis, we found that HIC1 binds neuribin through the PDZ binding domain, which is the last few amino acids of HIC1. And you can see here that YFP is reconstituted when you put half of YFP in HIC1 and half in neuribin, while if you remove the last 4 amino acids of HIC1, you lose that. So all of this indicated, we then looked more at neuribin, and we found that all the phenotypes that were conserved in HIC1 were conserved in neuribin. And the final experiment we wanted to do was to see if we were to add the actin binding domain of neuribin onto HIC1, could we rescue all the phenotypes that are associated with HIC1 and neuribin? And the answer was yes, we could partially rescue all the phenotypes by just adding the actin binding domain that was present in neuribin to HIC1 without the last 4 amino acids. You could partially rescue the aldecarb phenotype. This is the mutants. The double mutants show hypersensitivity to aldecarb. This is wild type. And you can see you partially rescue the phenotype by adding, the actin binding domain of neuribin onto HIC1. We also rescued the Wnt secretion phenotype. So our current working model is very interesting, where we have a claudin that sits presynaptically in the cholinergic synapses and affects the actin cytoskeleton through an actin binding molecule called neuribin, and does so by affecting Wnt release presynaptically. So why we think this is really interesting is that there are a lot of parallels between how HIC1 functions and how claudins function at the epithelial cells. At least intracellularly, they seem to be functioning very similarly, where this molecule binds to an actin binding protein and affects the actin cytoskeleton. And the same thing seems to be happening at the neuromuscular junction. So we are hoping that some of this can be taken forward, and we can see whether this actually happens in mouse embryos or mouse embryonic neuronal stem cells. So with that, I want to thank the people who've done it, Veena and Pallavi, who've done all the work that I've shown here, a lot of people who've helped us. Many labs have given us reagents, and Zita, whose lab in University of Queensland, has also helped us with a lot of the recordings that I've shown here. Funding, I want to thank the IndiaAlliance, Department of Biotechnology, and ICER Mohali. And I'll take 1 minute to just talk a little bit about how the Alliance has helped me throughout these last 6 years where I've been an Alliance Fellow. So essentially, when I started my India Alliance project, what I found was that my project was essentially looking at cell surface molecules at the neuromuscular junction and essentially figuring out signaling mechanisms of these cell surface molecules. And what I found was that what we found was a number of molecules that are involved in this process. I got to talk to great mentors who gave a lot of suggestions. That really helped. But more than this, during my third year, we moved in a different direction because of an undergrad who had found some very interesting observations. And what he wanted to look at was neuropeptides and how neuropeptides affect, allow for crosstalk between one neuron to another. And he had some very interesting observations, which I discussed with my mentors. And I was a little worried because this is not part of my grant. However, the mentors were extremely helpful and they gave a lot of suggestions. And we went on to pursue this. So this is the second direction that we have in the lab, looking at neuropeptides and how they affect other neurons. And we have one publication that's come out and a couple of others that are in the process of shaping up really nicely. And this was thanks to the mentorship that I received from the Alliance. So for that, I'm extremely grateful. Of course, I want to thank all the Alliance staff, especially my grant advisors throughout these years, who've been amazing. They've always replied to my questions very, very quickly. I also thank the Alliance because thanks to the Alliance and the flexible funding scenario, I was able to set up multiple collaborations in Queensland, the US, TIFR Bombay, ICER Pune, and NBRC Manesar, all of which have been extremely fruitful. And I think some of these are also with India Alliance Fellows, so that was extremely useful in many, many ways. A lot of my students have graduated or are in the process of graduating, and essentially I'm extremely grateful for the flexible funding and the amount of funding that the Alliance has given that has allowed us to go through all these projects. Thank you.
Thank you, Kavita. We can take only a few questions.
Hi, Kavita. Good talk. I just wanted to ask you, given that occludence junctions are known to be present in the neuromuscular junctions, in the context of your HPO1 story, have you ultrastructurally examined the NMJs of the elegant?
Yeah, we have not ultrastructurally examined because it's very difficult to do it, but that's something that we really want to do. Actually look at it ultrastructurally, and the other thing we want to do is actually see if HPO30 is just required to bring these different subunits together, then can we do this, mimic this in, say, a Xenopus oocyte or something like that? Then, that will then give us exactly what HPO30 is doing, and then look at the mutant. Because I have a feeling what is going to happen is, at a structural level, there might not be sufficient disparities. Because I think synapses are formed normally. If it's just looking at the receptor levels, it's not very easy. We're not going to be able to find it very easily from ultrastructural analysis. But I think that's something that we are hoping that we can do in the future.
Okay. If there are no more questions, we break for tea. Tea is served at Silver Oak at the opposite building.
Was that very long?
We will come back at 11:45 for a very interesting. It's at ground floor. Silver Oak is the name of the hall. We will come back at 11:45 for a very interesting topic of discussion. from University of Oxford. I request all the panelists to come up on the stage and take their positions. Another panelist is Professor Anne Ferguson-Smith from University of Cambridge. We also have Professor Sandhya Vishweswaraya from IISc Bangalore, Professor Jyotsna Dhawan from CCMB Hyderabad, and Professor L.S. Shashidhara from IISER Pune. I request the panelists to come over on the stage, please. Professor Roop Malik is with us. He is an IndiaAlliance Senior Fellow at the Tata Institute of Fundamental Research, Bombay. And he's a biophysicist interested in nanoscale molecular motor proteins. I request Roop to kindly moderate this session.
Hello, everybody. Welcome back. And so this is the second panel discussion following what we had yesterday at Vigyan Bhavan, which I'm sure many of you attended. And today we have people who have been in this business of building leaders and have led in many capacities themselves. And we will talk about what it means to be a research leader, how does one go about it? When does one, embark on this? So we have some kind of a brief outline which you can see behind you on the projection. And what are the challenges in becoming a leader? And now this becomes particularly pertinent to discuss this today because as we celebrate the 10th year of India Alliance, we all understand that the basic mandate of the Alliance is to develop research leaders. You all appreciate that, science is exploding in the country as everywhere else, and there are basically, IITs and IISERs coming up in your neighboring towns, wherever you are. So with this, large number of scientific manpower which is being developed in the country, we do really need a proportionate number of leaders and, people who will lead this effort. So then obviously the question that Professor Bhan, M.K. Bhan raised yesterday at Vigyan Bhavan during the panel discussion, where he mentioned that he went to a meeting of deans and directors where things were said and things were discussed, but he really did not see people coming out to take those leadership positions. So I think it is important we talk about this. And we talk about this in a constructive way, where we try to look for solutions, not just bemoan what is not right with us. And we also talk about some of the India-specific aspects of leadership. What are the, how does the ecosystem support leadership? And for that, I would also request, Shashi, Sandhya, and Jyotsna to pipe in at some point of time. So we will get started. We hope that this will be a very interactive discussion. That is something which we have all agreed to within ourselves. And I would request you to raise your hands, and I will take the discussion to you as and when appropriate. I would also request you that you can interrupt us at any point of time. And if you have some questions or comments, we would really welcome that. So then maybe we can start with what is it that what do you see as a leader? Who is a leader, and when do we start leading? Maybe I will start with you, Noel, to get your thoughts on that. What is your perception of a leader? But before that, let me just remind you one point, that we are in Delhi, and whenever I come to Delhi, when we talk about leaders, we talk about policies and directors and all those kinds of things. So this is not about that. This is not about leadership on that level, but leadership on a day-to-day basis where you basically advance things around you. So please, Noel.
Okay, so we started. So the art of academic leadership is probably the science of herding cats. It's not corporate leadership. We don't have command structure in academia. We don't have one person saying what can be done. We're in charge of our own grants. We tend to be opinionated people. So I think anybody who's been on the corporate management course, I doubt it has little relevance to what academic leadership is. But the question was really, where does it start? Well, I guess it starts, I think, really from probably the first group you have. I don't think it really starts as a PhD student, and I don't think it starts as a postdoc. But the first time you have a group, you have to lead. And certainly when I first started as a PI, there was no such thing as a training course on leadership. There are now training courses on leadership. I don't think they train you how to be a leader, but there are courses on this. And then your responsibility then as a leader is to yourself and to your team. You have to get to know how to work with people. Again, there may be your PhD students, but those of you who've had PhD students and postdocs recognize that you can't tell them what to do. In fact, if you can tell them what to do, they're probably the wrong student. Get rid of them. Your best students are the ones who don't do what you want them to do. So you're working with people, and this fuzzy line between leadership, by which I mean command, and mentorship, by which I mean persuade, that's what I think you have to transcend in academic leadership. You have to bring people on board. This goes all the way up to once you start sitting on committees, you end up having the power of persuasion. You do not have the power of command, even if you're a dean or a vice chancellor. So I think it's a very different style of leadership in academia to what you might find in the corporate sector.
Yeah. Maybe Anne, would you like to comment on that?
So yeah, absolutely. I think one of the most important aspects is to let the science do a lot of the leadership for you. So it's about having enthusiasm for science, motivation for science, and curiosity for science. And that aspect of your scientific persona should motivate and inspire the people around you. So there's this motivational aspect that I think is very important when you're leading a group. And then the second aspect is really, it's all about the people. It's about the science, and it's about the people. And so it's about developing relationships with the people who you're working with, not only within your lab and within your group, but your immediate colleagues and your collaborators more widely. And it's developing a set of standards that you feel important to you to live by in that kind of context, which is about respect, and it's about communication, and it's about, I think, also having a spirit of generosity. Sharing your science with other people, motivating other people and inspiring other people about the science that you care about. And I think if that little package comes together comfortably, then everything else kind of follows naturally in its place.
Great. So in that context, perhaps one of the questions that comes up is that, to be recognized as a leader, you need, in the academic field, you need respect from your colleagues. And that probably to some extent comes about when you have yourself achieved something scientifically, so that your colleagues, your younger generation see you as somebody. So maybe, so now this you have to balance your own academic achievements with taking on greater responsibilities. So would Sandhya, you would like to comment something on that? What's the best way of?
I think there are many of us for whom the prime motivation for being in science is to do good science. And I'm not very sure that we think too much about taking on leadership in institutions. We don't factor that into our thought process. We just want to be good at what we're doing and possibly being recognized for the science that we've done. In the context of then having to be forced to take on an administrative responsibility at a later stage in one's career, there is this very difficult decision that you have to take in terms of how much time you can give to your science and how much time you can give to administration. And I admire people who are able to do 50-50, but I think you, at some point, are compromising on one or the other. And if you decide to take up administrative roles, there is going to be a compromise on your science, and you're going to have to live with that. It's a decision that you take, and most of these decisions are taken at later stages in your career. And I think most of the audience is below 45, maybe 50, and perhaps these thoughts aren't even entering their heads right now, and they should not. This is the curse of old age, that at some point of time, you may be asked to take on responsibilities that will take away a little bit from your science, maybe not a little, maybe a lot. And there are some very influential administrators of science in this audience as well. And I'm sure all of them have made a conscious decision to give up something for the sake of perhaps in their view, only in their view, a greater good.
So I think that takes us nicely to Shashi. So Shashi, when does one start to get into these decisions of policy making, doing things beyond your lab? What was your own experience? Can you tell us something about that?
I don't know. Sorry, I think I agree with Sandhya. to start with, again, the leadership is not about governing, administrating, or, as you said in Delhi context, ruling, other people's life. I think leadership is about ourselves, our own life, and what is that you want to do, how you want to do it, and your own terms and conditions is what is the leadership is all about, and trying to see whether, the system can be engineered to help you, right? Now, in my life, I decided if I want to contribute to science using my own, personal research and with the help of PhD and postdoc, if I can contribute to some arbitrary unit of 1.0, and by facilitating others to do good science, if I can contribute to science, let's say 1.2, right? And maybe it's worth going to that direction. Being an evolutionary biologist, I think that has a higher fitness than my personal science. So, and I think I decided at some point, at much earlier age than you were referring to, below 45, when I said, okay, maybe it's worth trying to do something different and help others to do good science. And I don't know whether it's considered as leadership because I always use the word facilitator, never use the word head of the department, coordinator, or dean, or anything of that kind. So that's one aspect. The second is in Indian context. We can have three different categories of people where people want to pursue their own science and what might come. And the second is help others and also build the institute and help others to do good science. And we need to ensure that when they come with some idea that I want to do something, rather than would you please do something, that's where you distinguish who has potential leadership. Potential leadership will say, I want to do something, rather than keep on giving suggestions saying that, system should do something, people should do this, institute should do this. Wherever people have come with suggestion clearly, or not suggestion, with a project or a proposal saying that I want to do something, either for school education, for undergraduate education, for research, or for outreach program, or just building a cultural ambience of the institute, we have supported that. So that's one way of identifying the leaders and supporting them.
So maybe Jyotsna, over to you. How do you think things have changed over the last 20, 30 years in this context? Has this definition of leadership changed at all in the ecosystem that you have seen?
So maybe I'll back up a little bit and just say something about an aspect which I think is inherent in this whole discussion, and that is that leadership is an activity. It's not a position. It's not a designation. And I think too often in our context of showing progress as we go through our so-called career advancement, there's a tendency to fixate on this designation part and not on the activity. And the activity is one which is personal, and each person comes to it with their own struggle and their own experience. And I think that is something that we need to focus on much more. And I think India is not different from anywhere else in this. Science is an intensely competitive activity. And our young researchers, I think, respond very strongly to that competitive aspect as, making certain benchmarks. But actually, the competition should really be with yourself, which is you at a certain stage and you later on in an understanding of a problem. So I think the leadership really is important in communicating to people who you work with and who you have the privilege of guiding in their PhDs or postdoctoral work, that it's actually important to sustain yourself from responding solely to the competitive aspects and really working on the science, right? And I think that is something that if you can set that ethos in your lab, and I've seen some who are very successful at that and some not so, and I think it's going to change at different times in your career because you too are responding to stresses which are external and many which are internal, because science is a self-driven process. So in terms of what's changed, I think there has been tremendous change over the last 23, 24 years that I've been doing this. One of the things that has changed dramatically is not, and it's not specifically leadership issue, but it's an issue which affects all of us. And that is, I think that the demographics of students who are coming to our labs, and this is an India-specific thing that I'm talking about, has changed dramatically. 15 years ago, most of the students who were coming to our labs were from the more elite urban institutes. Now we have students from everywhere. So I think, and this is a very, very positive thing, right? But it also means that there are cultural issues that have to be dealt with in working together as a group. Most of it has been dramatically positive. So I think that this is a really great thing. But it also means that a leader of a group has to be sensitive to interpersonal relationships within a lab and build things that are actually encouraging to everybody in the lab and not just catering to a set of people who immediately understand what it is that you're saying. So that, I think, calibrating yourself with everybody's capabilities is something which is important. So I think I went a bit off the mark there, but...
Well, thank you, all of you. So would the audience have any comments on the things with respect to this issue? What do they think is leadership about, please? We'll keep this slightly short because we have to get on other things, but is there a mic
somewhere? First of all, I would like to introduce myself. I'm Dr. Aman Gupta. I'm currently associated with Oxford University, doing my specialization in sleep medicine with Nuffield. And so my question is, to Professor Jyotsna. Ma'am, I want to know your opinion, how the top-notch universities and institutes, they help develop leadership. You spoke about, you are now getting students from different parts of the, Indian, earlier you were having the top-notch students, now you're getting, the students as well. But leadership qualities has something to do with an institute you have been working or being trained into. I just want to know your thought on that.
So if I understood correctly, what you're interested in is how have the institutes with which I have been associated recognized and developed leaders? Yeah. So, every institute has a structure which recognizes people who achieve scientifically, right? And so that's what I meant by the sort of marks that are along the path. Now, some of those are deserved, some of those are not deserved, right? But let's not get into that. I don't think many institutes have a strategy for identifying leaders. So, and I think this is something which you can't legislate. It is something that grows out of the culture of individual institutions. Perhaps what one can say is that it's important that one takes into account not just the the awards and the rankings that come along to a person, but really, how are people doing in their labs? Have they identified deep problems which are important and are going to, they may take time to develop? How do they interact with their students? What is the culture of egalitarianism that they sort of create within their environments? Are they people who are going to, as Shashi said, step up and do something without just complaining about an issue which they can see is systemically a problem, right? So I think every institute deals with this in different ways. But I will say, yeah, Shashi has something to add here.
So one easy way is, leadership is also like an expression of something, right? It's like a creativity. So you give them an opportunity to express. And more the number of times they have an opportunity to express, a true leadership, maybe be able to be recognized by the other leaders of the institute. So one way is democratizing the entire process of the institute or the organization so that everybody has something to say, depending on who says what thing, how they, what they say, how much they practice. It's an easy way to identify the leaders. If you don't have a democratic setup, you'll only see the so-called deans and heads of the department. You will not know who else would be the next, potential leader.
So one of the things that goes with leadership is responsibility. And with a very good leader, responsibility and vision. It's not just being in response mode. So I think responsibility is a very important part of leadership. And to train people in leadership, You start by giving them responsibility for things, maybe little things, and then maybe slightly bigger things, and then slightly bigger things, such that they develop comfort in taking responsibility and confidence in taking responsibility. And then and of course some people don't like it and don't want to take responsibility, and they make a choice to not take leadership roles. But those who are comfortable in it and like it and do it well are the ones that you nurture and integrate into succession planning opportunities. So unfortunately, with responsibility often comes more responsibility. So that's sort of how you develop a leader, I think.
We can take a quick comment from the audience. Yeah.
It's been implicit in several of the points that have been made earlier, but perhaps it would be helpful to make explicit that in science, leadership works at many different levels. And although there are common features and attributes, what it takes to make a good head of a lab may be quite different from what it takes to lead an institute, may be quite different from what it takes to lead policy development at the government level. And perhaps in the ensuing discussion, that those types of differences might helpfully be, touched upon, because I don't think there's one common set of characteristics that would work at all these levels.
So since the mic is with you, why don't you give us your thoughts on that?
I think the thought that I had was what I'd expressed, which is it takes very, very different personae, very different features, and very different attributes to be successful at these different levels. There are clearly some commonalities. I think as Noel and Anne and Sandhya as well alluded to, the difficulty in leading in science, it's not just herding cats. It's about how do you lead a creative enterprise where the products are not that clearly defined and the routes to make your so-called product, the endpoint, are not defined at all. I think it's, this is what makes the whole thing complex, and I really don't have, anything to say that would characterize what you'd call a good leader at any of these levels. My experience has been there are people with widely differing personalities and differing skill sets who are extraordinarily successful in all of these things, leading labs, leading institutes, and in influencing policy.
Would you like to add to that, Sandhya?
Maybe a little bit of a digression, but I think there is one common core that perhaps all leaders and presumably all human beings should have, and that is empathy. And I think we must look at all the people in our environment, whether our students or our colleagues, as being individuals. And we must make an effort to try to understand them as human beings first, and then later on cater to other needs and requirements for the scientific enterprise. And there is no good leader who doesn't have empathy and responds to their surroundings or their environment. And the way that one responds depends on one's own character. Let me quote Shakespeare here: "This above all else, to thine own self be true." It is extremely difficult for many of us, and especially scientists who believe in truth as being their driving motivation, to compromise on aspects that they believe to be very, very strongly, important to them as human beings. Ethics, ethical approaches to whatever you do, whether it is your science or you're dealing with another human being. These are all characteristics which are going to influence the way that you deal with situations. And you can't take the human element out of any decisions that one makes as a leader.
Noel?
Yeah, I don't think we should lose Ashok's really important points, especially about the different types of leadership which require different roles. But I do want to go back, and it was prompted by the question from the front there, which I think was aimed at what are the institutes doing for leadership. So as coming from the outside, I want to do a bit of audience participation here. So you're only allowed to put your hand up sort of towards this question or keep it down, but you've got to give a bilateral answer. Who here has had anything that they think approaches useful leadership training? All right. Who here thinks they have not had adequate leadership training? Okay, still a lot of people sitting on the fence, but most people have not had it. Thanks, I just wanted to know that. But this is one of the problems. I think probably it will be a separate area of discussion. What constitutes training? What can you be trained? What can only be learned by exam?
Maybe we take a question from the Rashna there.
we've been talking a little bit in abstract terms, and I think I really appreciate what Sandhya said about empathy being a quality in a leader. That's really important, because whether you like it or not, every lab head is a leader and a mentor. And I think it would be very naive of us to think that every great scientist can also be a great mentor and a great lab head. But one never hears anything about testing leadership or mentorship qualities during recruitment. You put in a project proposal, you get your echo letters, and you're appointed, and then said, okay, here's the money, get your students and start. Can that change? Can we actually look for leadership qualities during recruitment? Is that happening anywhere?
I mean, to some extent, in some places, at least, we try to practice in our institute and also in the committees in different institutes when I was there. Sometimes this discussion comes. How good is this person a mentor? There's usually in the CV they have to mention, have they mentored undergraduate students or master students? They would, because usually they come after postdoc. It's very difficult to have, because they would not even had an opportunity to mentor others, right? So, ability to collaborate is one way of, reflecting on their mentorship, because how well you can collaborate with others is also a reflection of to what extent you can, interact and socially and in a very friendly way, in a very productive way. And that's somewhat a reflection of good mentorship too. But there's no really ideal way of identifying unless you put them in the field, jump into the akhada and then see, what will happen. Sorry.
We'll request Anne to.
It's not that hard to actually integrate that into application processes. The Wellcome Trust, certainly, being somebody who applies for grants to the Wellcome Trust, has sections where you have to fill in your information about who is in your lab and how you run your lab and how many people you have and who you've trained and what they've gone on to do. And so there's a section in the application that the reviewers read that looks at your track record in that area, and it doesn't take up a huge amount of space, and it's something that I always appreciate and read when I have grants to review. It tells you quite a lot about the PI, and it's not a difficult thing to do.
So I agree that that's probably a useful exercise somewhere down the line, after you've had your job for a bit. But responding to Rashna's question, which was, can you recruit based on some signs of leadership? I think it's very difficult because, my own experience of being a PhD student and then going on to be a postdoc, when you get your lab for the first time, I think one has no idea when you walk in that door of all the disparate activities you're now going to have as your responsibility, which you simply didn't realize your mentor was doing while you were a trainee at any level. So I think that in itself is such an enormity that to think of yourself now in this position of leadership is, I think, for a committee to sit there and recruit based on, these signs of leadership would be difficult. What a committee can do is see the personality of a person in terms of how they express themselves and how their scientific sort of creativity and persona comes out. But I'm not sure that, their leadership qualities are so apparent at that time.
I think that's right, and I think it's very easy to make mistakes. And I think that mistakes are extremely useful things to make. And it is unfortunate that most of our training is done from learning from our mistakes. I think it's very hard for there to be rules, in fact. It's about resonating with a person. It's about making sure you do your homework and asking about this person. But also you're going to make mistakes, and actually that's all right so long as you look after the people properly.
I think
so
something that
I
I would
radical suggestion to that. You always have to say whom you've mentored. How about reference letters from those students you've mentored?
That's exactly the point I was coming to, that when you write reference letters for people you have trained.
You can set a benchmark for the future career so that the people will actually realize that there's also part of their career progression. For example, all the major recognitions like fellowship of academies should be largely based on the mentorship, not just only on the publication.
And I think also when we, write letters for people who have worked with. This is something which we should, dwell on. What leadership qualities did you see in this person? We can.
Yes, I think, Rashna, you have a very good question, and I had the most eye-opening experience very recently that is related to that. After spending 16 years at Johns Hopkins, I went through a series of interactions to move to Mayo Clinic. And in no uncertain terms, they told me that if you want to be a star here, this is not a place for stars, but this is where we have constellations. And all of their background checks over 1 year were aimed at whether or not you're a team builder, whether or not you're a team player. It was very, very important. And now that I'm there, although for a very short time, less than 3 months, I can see that this is a place like almost no other place I've ever seen, where the focus is on people working together and trying to solve a common problem. And they actually have leadership courses. They have administrators whose sole job in life is to help the academic leader. They run an academic institute almost like a corporate enterprise. So in some ways, they take the best of corporate world and then they bring it into academic ways. But they are very highly selected. There are very few positions. But it was refreshing for me to see that they want to not go wrong in that recruitment. And they are going to take all the time they want because they want the right people who can interact with people in the right ways. Although they don't go so far as to get letters from your trainees, but they are doing enough investigation to say, are you going to be a good member of the community? And I think by extension, this is not a true test of leadership, but it's a test of being a good member of the community because you're going to build future communities.
I think that brings us to a point which I would like to take up now, coming to the Indian context. And who's that?
Yeah, so
which brings me to the Indian context. And because Noel, you have been associated with the Wellcome Trust and have seen now over the years how things work or don't work here. What do you think we should do differently? Should we? How do you think this leadership, training quote-unquote leaders, what do we need to change here?
Well, in a sense, I'd probably abrogate that responsibility because I thought about this before. And if we think about what the job of the Wellcome Trust has been, certainly for the intermediate and the senior fellows, it's been, in the case of the intermediate fellows, to recognize potential stars, and by which we mean potential scientific stars, and the senior fellows recognizing people who've already achieved that. It's not directly, I think, about honing leadership skills. We do hope that those people will go and be leaders, but as funders of fellowships, it's not really where the training comes from. I'm not saying it can't come from. I'm not saying there's something the Alliance can't do. But leadership is specific to an organization. It depends on the context and the culture you go into. So I think the leadership coaching, training, and practice is something that must be provided by the host institute. It can be enabled by organizations such as the Alliance. But we, I don't think, are really trying to recognize, I mean, Shahid may disagree with this, but as the chair of the committee, I don't particularly look for somebody who I think is going to be the next chair of faculty or anything like that. I'm not sure we're equipped to recognize that. We're looking for people who will be scientific leaders, and that might be a different distinction. But those skills, those personnel skills, perhaps the sorts of training that was being alluded to by our previous questioner there, these should be provided, I think, by the host institute. Maybe the Alliance can do something about enabling that discussion, but I don't think it's our job as electing fellows to actually really be responsible for that duty. So I'm not trying to abrogate it. It's very important. There's a difference between suggesting people for roles for scientific leadership in a particular domain and recognizing how they may provide leadership within an institute or a university.
Jyotsna, would you like to pick up on that?
Yeah. A little bit sort of in reference to Akhilesh's question as well and building. I mean, from an institutional perspective, I think it's very critical that an institute's director or leadership recognizes that there are going to be people who are scientifically astute and fantastic scientists, but are not going to fill all of these qualities that we've been talking about here. I mean, I'm talking about the cranky loner who wants to just go ahead and be left alone. And I think institutes really need to respond to leaving such people alone and not herd them into participating in certain things. Having said that, I think it is important to recognize that there are qualities that anyone can bring to the table and participate in enabling an institute to grow. But I think in most Indian institutes, at least the ones that I'm most familiar with, there isn't really a dedicated process to looking for scientific leadership. And I think that we could do with a bit more explicit thought, at least. If not, the trouble with having these kinds of things is that they get very quickly prescriptive, and that should not be. It needs to grow organically within the culture of an institute, I think.
So, I somewhat sort of disagree with Ashok to some extent. The idea is, see, India is moving from so-called service economy to knowledge economy. Now it's fourth or fifth largest economy, but our R&D community is several times smaller compared to Europe or USA. And we are expanding. And in this expansion, we had to bring in a completely new way of governing our academic organizations, which is very different from the 100, 200-year-old bureaucracy we are all very familiar with. So in that context, I think what Noel said, I think training is the most important thing. We need to identify leaders. We may have to make some compromises and train them and make them to understand the nuances of academic leadership and then trying to bring them. We cannot have enough number of natural leaders who can take up academic leadership in this country. I think we may have to proactively make some people to understand the requirement and train them to take up the leadership program.
If I can just add something, I think the kinds of leaders that you need are going to change very dramatically over the next 10 to 15 years, as they have changed if I'm talking India-centric right now. At the time of independence, perhaps the leaders that were required were those that could make decisions and get jobs done, because the country was in a sad state and we needed to build institutions, we needed to set them up, we needed to make sure that science could progress in the country. With the generation that we have with us now, and I'm talking about younger scientists like you and of course our students who are one generation below that, there's got to be a participatory way of doing leadership. It is not possible now to make decisions on your own, not with social networking, not with input coming at you from all sides in so many different ways. You cannot be dictating terms anymore. And so it's, as I said, it's an evolution of the kinds of leaders that are required. And you have to be sensitive to that change that is going to happen and which we ourselves have seen in the last 20 years of doing science in this country. And unless you're receptive to that change and are willing to adapt to it, it's very difficult for me to say today, this is what's required for leadership 10 years down the line. The rate of pace of change is so much that I don't even know that I can see what it's going to be like years down the line, let alone 10 years down the line.
So yeah, I wanted to sort of follow up on that a bit and also think a little bit about your comment about a constellation and in reference to Roop's question about what the Alliance was doing well. And one of the things that I've really enjoyed watching grow over the years has been to see the network of fellows that have evolved as this program has got bigger and more inclusive. And this constellation of fellows, if you like, is the future of science in India. You can't make things happen on your own. If you're a leader and you're going to make things happen, it's about making things happen with your network, with your colleagues, through communication. And it's this very type of network of fellows that really is going to allow this to happen. You know each other. You're going to, as you develop and grow as scientists and as and in maturity and leadership, you're going to be reaching out to each other to make change and to make things happen. And that's, I think, one of the really important things that this alliance is doing.
Thank you, Anne. We have some kind of, yeah. Can we have a mic here? Okay, please, go ahead. Why if you have the mic? Go ahead.
So from the point of view of somebody who's aspiring to be a future, like do their research and develop their own group down the line, what, from your extensive experience that the panel has, what could be a possible advice that you would go back and give yourself, that these were the learning opportunities which would have helped me to become a better leader in future for us, so we can implement those things today, and so it is helpful for us in the future.
Okay, who wants to take up that challenge?
So in one sentence, all your bad experiences, don't pass it on. All your good experiences, amplify and pass it on.
Okay, great. So
just a specific example, when I did my PhD in Cambridge, my supervisor, PhD supervisor, and extremely, good, and she gave me full freedom to do what I want. And then when the publication came, right, and as a PhD student, even in 1990s, anywhere in the world, it was very difficult to be a corresponding author, right? And I was the corresponding author of my very first publication for my PhD, and that sets my life. Because all my future jobs and everything was dependent on how good my publication was, right? And my very first publication, because I was a corresponding author, in addition to being a first author, it's basically made me, have a quick jump start. And that's because of the good mentor.
Exactly. So I wanted to add that his supervisor showed excellent leadership skills, right? She could see the potential of a future leader. And as far as most of you in the audience are concerned, I think right now just focus on your science. The rest of it will take care of itself. I think if you get too involved in some of these thoughts right now, it may not be worthwhile. Because remember, this leadership thing is a pyramid, right? So if you aspire to reach the top, it's going to be very difficult because people are going to fall off along the way, but you need to be very happy with yourself and your life, even if you don't reach that pinnacle. So just focus on the science.
Great. So,
perhaps I could distill an admittedly provocative question which comes from listening to a lot of this discussion from my original point, which is this. So look, we select at the level of postdocs, fellows, group leaders, for people who do imaginative, original science and very little else. And from that pool, we are also then choosing the next tier of leadership, people who lead large groups of people, people who need to influence policy at government level. And we all admit that these skill sets are quite different. So we're training and selecting throughout the early stages of people's careers for one set of attributes, and then from that selected pool trying to find what, in my opinion, is a very different set of attributes. Is that the right way forward?
Maybe Noel, yeah?
I completely agree with what you say, Ashok, but I mean, I suppose the only pushback against that is that of the 100 people that we choose to be successful postdocs, to be successful PIs, you only need a very small handful to rise to that next level. I'm not even sure what's rising, actually, but to change that next level. And the thought I'd put hand in hand with that is, having been on the end of various management courses, usually compelled by different universities, and I've not been convinced that there is much trainability in this. I think the thing that we're missing here is a lot of the reality of leadership, certainly at the highest level. Those people who can really have the breadth of vision, those people who have the force of personality to take people with them, again, not by command, but by persuasion. I think a lot of this is actually an extension of personality. It is your personality. So I'm not saying that there's nothing you can do to enhance people's leadership and management skills. I think you can do that. But the sorts of people that I think you were probably referring to, I think they will rise out of a sea of talent that we have there. Not everybody has to do it. In fact, if everybody has to do it, we'd probably have open warfare. So.
Yeah. We'd like to go back to the... okay, please, go ahead.
Yeah. So if I can pick up on the thread of empathy and maybe challenge it a bit, isn't leadership to some extent a matter of balancing empathy? Because as leaders, whether we sit in judgment over the lives of people, over the lives and careers of people. As professors, we sit in judgment over the careers of students. As a leader of an institution, of a funding agency, of a corporate entity, we have to balance the empathy for the individual with the larger interest of the organization, the larger interests of the science. Studying what traditional, in the traditional picture, people have, those who have been considered leaders, basically, kings, military leaders, and things like that. And it's probably more a lack of empathy that has propelled these individuals forward. And the other thing is that I'm not sure that we can. It's a good advice to say that leave such thoughts until you are 50, because the art of persuasion is not something that is learned when you are 50 plus. The art of persuasion, of taking people along with you, which is what constitutes a large part of leadership, is something that from the student days, those who are active in organizations, in students' unions, and volunteer activities, they express these leadership skills all the time.
So I'll just mention one sentence, the empathy part. The institutions excel only if individuals excel, right? Institution and individuals cannot be separated, right? Finally, institution is just a bunch of individuals. So empathy is about actually individuals. And let's say you have to cull someone because it didn't perform well, right? But you can still use, appropriate ways of doing it and relocating that person so that the person, when they leave, the person also leaves with certain happiness and understanding that that person didn't fit very well with the institution and its aspiring aims and goals. And that's one way of, and building an institution and maintain. But there is nothing like balancing empathy here. It's just, you have to be empathizing with the individuals, then only you can, build the institute.
I just have to respond. Half the problems in the world arise because leaders don't have empathy. If you just think about some of our political leaders out there right now, not necessarily in India, the problems are arising because they don't have empathy. So when I mean empathy, I don't mean sympathy. I mean empathy in terms of understanding another human being. And as Shashi correctly pointed out, yes, there may be some hard decisions that you have to take. But how you present that to the individual, you do based on their character and their personality. That's what I meant by empathy. The second thing that you said is about learning skills of leadership from very early on. It takes a long time for us to be able to persuade our children how to do things, right? So we grow with them, and it's maybe by the time you're about 40 or 45 that you feel that you're on the same plane as your kids. So it does take time to learn this ability to be able to persuade and convince people around you. It's not that easy.
Thank you, Sada. We'll go to the lady at the back there. Yeah.
Hi, thank you. My name's Anna. I'm here from the Wellcome Trust. I missed the very beginning, so I apologize if this has already come up. But and following on from the last couple of comments, actually, I'm struck by the slightly strange gender neutrality nature of this discussion about leadership. I was at the Women Leaders in Global Health conference last week that Wellcome sponsored in London. One of the things we discussed a lot was the fact that of the global health workforce, 75% of those people are women, and of the people in leadership roles in global health, around 23% are women. We don't live in a gender equitable world, unfortunately. So I think it's important for a discussion like this to acknowledge that, and I'd be interested in thoughts from the panel about enabling women leaders in science specifically? Thanks.
Jyotsna, would you like to take that up?
So I think it's very important that we do that. And it needs to happen at a very early stage, because I think, as you well know, one needs to see role models in order to even aspire towards goals that might have seemed completely off your radar. I think it's important that we do that institutionally, because, for example, one of the things that I think we, in many institutes in India, don't do well is acknowledge the fact that we don't have a gender audit of our institutes, and that we don't have recruitment which is based on that understanding. Invariably, faculty search committee discussions sort of devolve into a question of merit versus gender, and that should not be. So I think there needs to be wider conversations on these, and we do need to acknowledge that fact. So,
thank you, Jyotsna. We'll take a question from the audience. We have to start winding up now. Please, Anurag, yeah, we'll move to you next.
I think we've had such a good panel, so I thought I would throw out a tougher question which is more brutal. Because so far we've been talking about things like gender neutrality, empathy.
Anurag, can you speak
where
up?
Organizations are supreme. The leader is there to serve the organization and take it forward. I was thinking of the Max Planck model. In the Max Planck model, as you all know, when the leader retires, they look for the best person in the world. If they can't find the second best or third best, the entire group can be dissolved. That's a very brutal model based on the fact that organizations exist to solve a problem, not for themselves. What do you think of that type of model of leadership that has served effectively, at least in one system?
You can take that to Shashi and Noel following that.
Well, I mean, I guess, that model has worked very well, right? So when in the context of, let's say, only look at the science as just, something which needs to be improved, something needs to be discovered, so all the time inventing something. So you're dealing only with that aspect, and then you want to really reach the excellence. As a society, what are the different models are possible? You try different things, including the Max Planck model. But I think Indian context, we are still at a very, very different stage. We need to make first science sort of pervasive thing of our personal life, which itself is, very, very, has not happened yet, right? We consider technology as science, and then we think that we are all, scientists. But really, the science and scientific temper has not really come into our, day-to-day life. I think at some point, I mean, I'm talking about the professional scientists. I'm not talking about the general public here. Unless that happens, and I think, Max Planck model is too early for us at this stage.
Noel, would you like to?
Yeah, in deference to any German colleagues who might be here, I don't think the Max Planck is the best model around. I do have some experience. I worked there, admittedly, a long time ago. The reasons for my critique are exactly your own point, and it really reflects the fact that in the MPI system, the director is king, the director is God. Everything revolves around that person. There's not much. I mean, I think things possibly improved in years, but I want to just make sure that this is not seen as a sentiment against the way the Germans do things. I've been really impressed, for instance, by their stem cell institute, which is a different model. And this is a model where young researchers come in with a 5-year program, and they're given good institutional support, quite generous support, I think in the order of about 200,000 euros a year, where they don't have to apply for grants, so they can go out and do blue sky research. And it's more like a skilled cottage industry rather than the Max Planck model. So I don't like the centrist approach that the Max Planck model gives. I would not be an advocate of that, because I think it stifles the individual juniors and stifles the curiosity lab and makes it all about the personality, which is why you get to this point when the Max Planck director retires or dies, and therefore the whole group goes down. So reasons not to like it.
So I think we'll have to start winding up, but we'll take some questions from the audience, please.
Listening to the conversation, I wonder whether a lot of the confusion resides in trying to separate management from leadership. A lot of the responses that I heard was about management in a fairly static, slow-moving situation, where leadership really is about having a conversation with the future and maximizing the potential of every individual that you're touching, and maximizing the potential of an institution and knowing when to make what changes to meet the future demands. That requires a completely different skill. It's a conceptual skill rather than a trainable skill in most part. And I think this is where I think that issue of empathy becomes extraordinarily important, where you're empathizing with the people around, with the future and the future generations, even those that you're not seeing directly. So bringing back to that, what aspect of that can we cultivate? And how do we capture the diversity of personalities? We talk of diversity in terms of genders, but there's diversity of personalities that need to come into this leadership mix. How do you do that?
Maybe a quick comment from Shashi. Yeah.
So we are a reflection of our society largely, small institute or large institute or university. So to some extent it's true that leadership of that kind you mentioned, and Josna also mentioned in the very beginning, there's a continuous process, is an activity which is not like one-time deanship kind of a thing. So in that context, it's true that a good leader is the one who thinks ahead and brings people and to take them with the dream for the future. But this is still trainable. It's not so much everything is creative. It's simply because we are still dealing with a system, dealing with a social endeavor, which is not something natural here, right? So it is something which is culturally evolved and not something biologically evolved. So in a way, everything culturally evolved is trainable, right? Like communication skills, language skills, right? How to communicate is trainable, right? And leadership also is a trainable kind of a feature.
Just very quickly, this is a very personal point of view. I think what you're talking about there is what I was mentioning before, which is about vision. And the best leaders are visionaries. And actually, I don't think you can train somebody to be a visionary.
We'll go to the audience there.
I was wondering whether the extent of freedom which is given at the level of the training, whether it's at the PhD or a postdoc level, that factors in developing you a better group leader and obviously a leader way ahead in the hierarchy.
Josna, would you?
So I think it definitely has an impact on your ability to freely express your ideas, and if you're given that. But you also have to take that to some extent, right? Because science is that type of activity. Even though you might have institutional structures which don't necessarily explicitly enable those things, there are ways of conditioning your environment. And I think this is something that is very critical, especially for young PIs starting out, and even postdocs and graduate students. One needs to understand that it's possible to participate in things that are going on in order to actually condition your environment towards something that you enjoy as well as want to have progressing. And so that means a certain level of dissent and taking the risk of speaking out. And I think that that's important.
Okay, Vaskar, you had some comment?
I actually just want to make a comment on leadership. So I think that from our group here, the question of leadership is for a youngster is what kind of leader would you want to be like? And so that's the role model you're looking for. And in many ways then the Max Planck model is a model of extinction. That leader doesn't exist any longer, right? So that's not a valid model, right? In my personal experience, I've been very lucky actually to have many role models of leaders, and I've had a choice of saying, who is it that I want to be like? And the person I chose was a woman, okay? And the reason I chose her was, for one reason, is that I realized that long after the students had gone and they'd become famous professors, whatever, outshining this leader, they still came back to pay homage. They still came back to say, ma'am, how are you? Okay, and how was life? Okay, and that was because of the empathy she showed. Okay, and that's the kind of leader I wanted to be.
Thank you. So we'll take one more last comment from there before we end.
Hi. Sure. Yeah. I just wanted your thoughts or experience. Anne had hinted at it slightly about learning from mistakes. But in our drive to just look at success and in the ladder of leadership, what is the importance you give to failure, recognizing it, accepting it, and coping with it and learning from it?
I actually don't think any scientific enterprise is a failure. Whatever doesn't fall in line with our hypothesis is teaching us something along the way. So I kind of dislike this word failure, and this, you know, I don't let my students come into my office and tell me, Sandhya, the experiment didn't work. I don't know what they mean by that. I mean, if you've done the experiment, there's a result, let's interpret it. And maybe it didn't agree with what we thought would happen, but that's okay. So I think something, you know, using words like failure are not really good for the way you think ahead. Take it as everything being a learning experience, and then I think you'll be on track.
Okay, we'll go for one last short comment there. And
very, very brief. So I think there was an excellent discussion, I think, but at the end of it, the question of how you define a leader is a bit abstract. I mean, it doesn't necessarily have to be associated with an administrative position. I think a leader, there are people with personalities which show glimpses of leadership right early on, even as a PhD student, as a postdoc, because I believe the fundamental common abstract point is that one has to have initiative, take initiative, and basically help people. I mean, that could be your students, it could be your next door, you know, lab, postdocs, or could be people in your own department. I think it's, and while you're trying to develop your own skill sets and develop your own position in the scientific community by getting recognition or publishing well, to set an example, I think it's pointless that if one were to just focus centrically on yourself and not probably help the entire department or the entire community. I come from a system where we leaders, that's a university system where departments' chairs are decided by rotation. I mean, the senior professors rotate every 2 years. There's really no system to select on basis of any objectivity like merit. So I think this is, and at times a lot of people who do not have those skill sets or are loners are forcibly put into situations where they have to take certain initiatives. So I think this is a great initiative by the trust to actually talk a lot about leadership. It's something which is not done in India that much, and I think a great initiative and more workshops like this will be very useful.
Thanks you, Deepak. I think we are running out of time, so I would like to thank all the panelists for and the audience for such a lively discussion and for interacting with us. I would say that key points I would take from this is, in very short, that leadership is an activity, and that we should look on to pass on the good experiences to our next generation, not the bad ones. And my own one is that if I can inspire the person next door to do better, then I think that I did a little bit of leadership today. So with that, I would again like to thank you all and for your time and to you all also. Thank you very much.
So before everyone leaves the podium, there's some breaking news, and it's really my honor and privilege to let you know that Roop has won this year's Infosys Prize. Roop, stand up and take a bow.
Thank you. So it happened on stage.
Thank you very much, Sandhya, Noel, Jyotsna, Shashi, and Anne, and of course Roop, for this wonderful session. We will move on to the next talk by Abhijit Majumdar. Dr. Majumdar is an IndiaAlliance Early Career Fellow, and he is also the Assistant Professor at Indian Institute of Technology, Bombay. His research is aimed at studying the role of mechanosignals in determining stem cell fate.
Good afternoon, everyone. So the norm of giving a talk is by thanking the organizer for the invitation. But I am not doing that today because शादी जब आपके घर में ही हो तो फिर दावत के लिए किसको शुक्रिया कहना. When the occasion is of your own family, whom should you thank for the invitation? So I am not thanking anyone of the DBT Welcome Trust for inviting me here, but lots of love, and please keep up the good work. So in this 10th birthday of DBT Welcome Trust, I would talk about this where physics meets biology. And whenever I talk on this particular topic, this also kind of my own journey. And I would like to share that today as well. So by training, I am a chemical engineer. And I did my PhD from IIT Kanpur with Dr. Animanshu Ghatak and Professor Ashutosh Sharma. And my work was on microfluidics, adhesion, adhesives, mechanics, soft mechanics, all those things, and nothing to do with biology. In 2010, when I joined Jyotsna's lab at INSTEM NCBS as postdoctoral fellow, there for the first time I did any cell culture. And these are the people, including postdocs and to the interns, JRFs, they actually literally kind of hold my hand to teach me how to do any biological assay. And I would like to thank Jyotsna here because not many PIs will take a postdoc to his or her lab who does not have any experience in that particular field. So I think that is some sense continuing with what discussion we have, that empowering people, and thank you, Jyotsna, for that. So when I say that I did not have any experience of cell culture, what do I mean? I would just like to give you a glimpse of that. So I joined Jyotsna's lab in 2010, 6th February, and this is from my scribble pad on 8th February. Unknown terms, mesenchymal stem cells. The terms I need to know, cell cycle, myoblast, cytosoluble myogenic cells, and motility. That is from the scribble pad of NCBS. I still have that with me. Starting from there, today, and just not because the knowledge gap, there was also the gap of language. I still remember very well once in Jyotsna's cabin when we were discussing, Jyotsna told me, stress and strain are two different things. I always thought I am stressed and I am strained are the same thing. And that day I realized that what biologists mean by stress is this, and as a chemical material scientist, what I mean by stress and strain is this. So completely two different worlds. It does not end there. What they mean by XY is this, and what I meant by XY was that. So you can clearly see it is a two different world that I started with. And today, I am leading a lab who works on aspects of cell biology, and we are doing our own iota of contribution to biology. That is all because of the training that I got during my PhD, obviously, during my postdoc, and the help, the support that I got throughout this process. So as I said, that two different worlds, right? And the first time I kind of see that the connection between these two worlds, when I saw that, okay, mechanics is there a common feature. Mechanics that I have learned during my PhD also plays a work in biology. And there I could see that connection. So what do I mean by mechanics? Anything that is anything to do with force, push, pull, spring, dashpot, pulley, anything you do with force is essentially mechanics. And we obviously cannot talk about mechanics without paying tribute to this person. Yeah, so the question that my lab deals with and the central question of today's talk, that what is the role of mechanics, that is the force, in different cellular context. So cells, our body is made up of cells and more cells, and how many cells? An estimation says 37 trillion cells. And how big the number is? In our Milky Way galaxy, we have 0.1 trillion stars, meaning is that your body has 370 times more cells than the star you have in Milky Way galaxy. And all these cells, they need to work in beautiful synchrony so that we can all kind of live a happy and healthy life. And that is done obviously by signals. That there are the signals that tells, okay, now this reaction should go, this reaction should stop, and all like that. And when I talk about signals, any biologist will think of something like this. The chemical signals, the set of chemical signals. But what I am talking today is about mechanosignals or mechanical signals, which are force-based signals. So just to give you one example of force-based signal is that I am talking, my voice is reaching you through the sound waves. That sound waves are kind of bending the tiny hair-like structure cilia in your ear, causing the ion channels to open, and that helping the ions to get into the cell and starting the chemical cascades. So mechanical signals, mechanical cues changing into chemical signals leading to biological processes that we talk about mechanosignals here. Question is it that the mechanosignals are only related to this kind of specialized cells, or is it a universal process that every cells they experience? So this one particular paper from 1980, I considered that as a giant leap in understanding the process of mechanosignaling. What they have showed that any adherent cell, they adhere to their surroundings and they apply force. And what they have showed that like if you have a very soft kind of substrate and then you have the cells, the cell adhere there, pull inside, and as a result you can get kind of wrinkle that you can get on your skin. Later on, people have made these micro pillar arrays, really micron scale, like your toothbrush bristle, much thinner than that. And cells apply force, they bend those pillars, and from the bending, you can precisely measure how much force these cells are applying. So this is essentially the model that cells attach, apply force, bend. Question is, what is the biological purpose it serves? To answer this question, we need to understand that to measure stiffness of anything, the only process that we have to apply force and see how much deformation it causes. This portion is softer than this portion because I apply same amount of force and I can see this deforms more, but this does not. So cells also do the same thing, and they apply the force actually to understand how soft or hard their surroundings is. Still the question remains, what purpose does it serve? This paper in 2006 from Disher lab, I consider that is another seminal paper. They work with mesenchymal stem cells derived from bone marrow, and these cells can give rise to osteo-like differentiation, bone, muscle, fat, neuron, and cartilage, all different kind of differentiation. Till 2006, what we knew is that we have to give a certain cocktail of chemicals to convert them into these particular lineages. What Disher lab showed that if you not giving the chemicals, but if you keep them on substrate which matches the stiffness, you can control their differentiation. For example, if you put them on a very soft substrate that is as soft as brain, the softest tissue in our body, you can get neuronal cells. If you put them on substrate that matches the stiffness of muscle, the mesenchymal stem cells will become muscle-like. And if you put them on a very rigid substrate, obviously you can guess the answer by this time, they will become bone-like osteodifferentiation. So essentially, by controlling the mechanics, you can control the direction, function, and fate of stem cells. Since then, we have got a series, many different physical, geometric, mechanical cues that people have shown that affect cellular function, cell fate, and cellular behavior. So as I told you, in 2010, I joined Jyotsna's lab. In 2011, I wrote my early career fellowship grant. I got that in 2012. I activated that in 2013. That year itself, I got offer from IIT Bombay as assistant professor. I moved to, in 2014, I moved to my permanent position. And again, the WT Wellcome Trust, India Alliance, you remember early career fellow was a postdoctoral fellowship. And they kind of made their rules flexible to allow me to carry my grant there. And as a result, I could kickstart my lab in 2014. So he is my first PhD student, Sanjay, who is now going to graduate. So this was the lab that we started working on, and you can see we were actually literally building the lab at that point of time. So we were working with, again, the mesenchymal stem cells. The problem was that, like any primary cell, if you try to grow them in the lab, after certain number of cell division, they just refuse to grow. They don't grow further. Their growth become a plateau. The number, so you cannot get enough number. Not just the number, but also they lose their quality. Quality in terms of their morphology. They become senescent. Sorry for the contrast. You cannot see the color, but yes, trust me, we have, they become senescent. And they also lose their differentiation potential. Meaning is that if I take a late passage cells, they don't differentiate into adipogenic any further. So meaning is that they lose their stemness. So you can think that cells become old as you grow them in lab. That was the problem, because to do any research we need enough number of cells, and if your one vial of cells after every 6 months they get over, and that was a kind of a continuous problem. And particularly in India, getting cells shipped from abroad was a pain. Also, when we started to look into the literature, we realized that it is just not our problem, but it is a major problem in the field, particularly in the regenerative medicine. So this is taken from NIH. They said once removed from the body, their capacity to divide is limited, making generation of large quantities of stem cells difficult. That is, if you want to do any autologous treatment, you need large number of cells, but as because they go into senescent, you don't get that number. So how to solve that issue? That was our first question. For some reason, we thought, I'm not getting into the reason right now in interest of time. For some reason, we thought that instead of growing them on plastic petri dishes that we generally do in the lab, if I grow them on a softer substrate, probably I may get a better result. So my student Sanjay, he took up the project, and so we make a polyacrylamide gel, stiffness of which you can control. You coat that with protein of your choice, then you put your cells, and he cultured the cells on plastic and on gel parallelly to see the effect. What we have found that LP, meaning late passage cells, on gel, they maintain their morphology, whereas late passage cells grown on plastic, they lose. Also, for some, also you can see that the growth, the number of the cells, if you culture them on plastic, that become plateau, that stopped growing. But if you culture them on gel, they just continue to grow. Here, the difference here in terms of the population doubling is 9, meaning I am talking about 2 to the power 9, that is 500. So on culturing on plastic, if you get 1 million cells, on culturing on gel, you can generate 500 million cells. That is the kind of number we are talking about here. Not just in terms of the quantity, but also in terms of the quality, we have shown that if you culture the cells on plastic petri dishes, as I told earlier, they lose their adipogenic differentiation potential, but if you culture them on gel, that is maintained. So you can maintain the stemness of your mesenchymal stem cells if you grow them on stiffness of particular optimized stiffness that we have done in the lab. Also, we have understood, we kind of think, I will not claim that we have completely understood the mechanism yet, that this is the difference we are getting, because when you culture cells on plastic, they apply more force. yeah, and like when you culture them on gel, their contractility is kind of little less. So probably that helps them to maintain their stemness. Next, what the question we asked is, instead of, I mean, okay, fine, we have grown them on plastic and gel. What if I switch them? After kind of growing them for a certain duration on plastic and on gel, what if we switch them? So what we did, we cultured them on plastic for up from passage 3 to passage 16. Then the cells which were grown on plastic, we switched them to gel. And cells which were growing on gel, we switched them to plastic. What we have found was that cells which were grown on plastic, when you put them on gel, they don't do that well. They do a little better, but again they become plateau. But cells which were grown on gel for this 13 passage, now even they are on plastic, they still keep on growing and again maintaining all their stemness property. And here we are talking about a difference in 2 to the power 18, meaning is that if you start here with 1000 cells, here you will get 7 million cells, but here you will get 1.7 trillion cells. That is the number we are talking about here. Also, we just out of kind of curiosity, we wanted to know what happens inside the nucleus in the chromatin structure. So my student Rohit with Dr. Deepak Modi at NIRRH ICMR, we tried to see the location of X and Y. Now I know like what X and Y are. And we have found that if you culture them on plastic, these two X and Y chromosomes are kind of far away, but if you have them on gel, they are much closer. It is somewhat it is telling us that when you culture them on plastic, your nucleus is always under tension, and that is probably causing a kind of permanent change in the chromatin structure. Now, this observation, that is the change in chromatin structure and maintenance of stemness, are they there is a causation, or is it just a correlation that we have to figure out? We have not figured that out yet. Next, what we ask the question is that this observation, is it just true for mesenchymal stem cells, or is it true for other cell types also? For that, we have collaborated with Professor Rahul Purvar in BSB Bioscience Department, IIT Bombay, and my student Pankaj, he worked on this. We took the keratinocytes, patient-derived keratinocytes, and we did exactly the same thing, cultured them on plastic versus cultured them on gel, and we have found exactly the same result. The only key difference here is that optimized stiffness for mesenchymal stem cells and optimized stiffness for keratinocytes are not the same. They are different. So kind of you can think of it in this way, that when today you go to, you culture any cell type, company give you that cell type specific media, cell type specific supplement. You can think of here the cell type specific petri dish that I can offer you, where your cells will grow much better. Other than this work, again, because I have a little flexible funding, so I could kind of venture around here and there. So we have collaborated with Dr. Shilpi Dutt of Actrek, and this work we have done with her student Angha and my student Ketki and Pallavi. So this work is on glioblastoma cells, that is basically brain cancer cells, type of brain cancer, and she has a very fantastic system. So in case of your this glioblastoma tumor, the treatment is radiotherapy. So if you do the radiotherapy, irradiate, you destroy the tumor, but again that relapses back. And clinically it has been seen that the relapsed tumor is much more aggressive than the parent one. What do I mean by aggressive? They are more invasive, they are more migratory, they are more proliferative, and they are drug resistant. However, if we put this parent cells and the relapse cells and put them on plastic petri dishes, you don't capture this difference. You don't see this kind of difference. So I will not go through all the details, but I will just show you two set of results. These are the parent cells and relapse cells. We have done an invasion assay. You can see when the cells are grown on plastic, parent and I mean, relapse is slightly more invasive than the parent, but not any drastic difference is there. However, when we cultured those cells on a material which is as soft as our brain, we see that relapse cells are much more invasive than the parent cells. Not only that, we also found that the relapse cells are much more drug resistant as well. So I will switch gear here, and I will show another set of work that we have done. So as a scientist, I believe in curiosity-driven research as well. What we have shown you is just a goal-oriented research, but now I would like to also share a curiosity-driven research from my lab. So quickly, I will just go through quickly. Here, we started asking question that instead of having a material which is homogeneous everywhere, stiffness is same everywhere, what if I just bring something in, because our body is not like that. So what Akshara did, she just put a rigid glass sphere inside the soft gel. So these green things are the cell. So they are at the surface. They are not seeing the glass bead. And what we found is that on a soft gel without bead, cells are arranged like this. But on a soft gel, when you have a bead there, all these cells are arranged in the radially arranged like a sunflower. And this arrangement is 2 millimeter long, meaning is that 20 to 40 cells, they are all aligned and creating such beautiful pattern. If you do a actin staining, sorry for, again for the contrast, you get all actin stress fibers are nicely arranged. This is a mechanics-driven pattern formation. If you play anything with the mechanics, the pattern formation goes off. I'm not going into much details, but I will just show you a video how this pattern forms. It is a migration-driven pattern. It is taken over 48 hours. You see the cells align. They can somehow sense that the bead is there. They try to move towards the bead and forms such beautiful pattern. So you can control this pattern. So we have checked that this is not ECM type or cell type dependent. You can get. What we have also done is that instead of having one bead, now if you can put multiple beads or bead and cylinder combined, or you play with the stiffness of your material, you can actually create different kind of pattern. Why this result is important? Number one, in tissue engineering, just getting a cell type is not enough. Our body is made, everything is patterned in our body. So to control the patterning is important. Also to understand how patterning forms in our body is an important question. So with that, I would like to conclude that what initially I thought that two different worlds, they are actually two interacting worlds, and cells apply force that can control or that can contribute to their destination, function, and fate. I have not spoken about cell survival and cell cycle, but how mechanics govern senescence, differentiation, migration, pattern formation, drug response, almost everything of their life. That is what we have shown here. So I would like to conclude by thanking, obviously, now the funding agency, IndiaAlliance. If you have noticed, in my first slide, I have used their old logo, and this side I'm using their recent logo. So in these 5 years, not just me, also the alliance has evolved, and I have obviously evolved with them. Thanking to IIT Bombay for seed grant, DST SARD for two other grants that I have, and my group members. Thank you so much. Thanks for your attention.
Questions?
In the context of the experiments in which, you were able to get the late passage cells growing on a gel, you didn't mention the composition of the gel. Just a follow-up of whatever you will say. what happens if you were to vary the chemical composition of gel, keeping the Young's modulus constant, and vice versa?
Okay, so that's a very good question. What we have done, we have not checked keeping the Young's modulus constant but varying the chemical composition, but we have done the other way around. We have used a stiffer gel. So we have varied the Young's modulus keeping the chemical composition constant, and we have found that you need a particular optimum stiffness to maintain that stemness. And as I have mentioned, that optimum stiffness is actually cell type specific. Oh, sorry, polyacrylamide gel of 5 kilopascal stiffness.
Hello. Regarding your experiments where you grow the cells on different substrates, so I was not able to understand whether it's the surface tension of the surface that is leading, or it's the actual force, total force pressure. So would it be the same if you suppose grow the cell in a different pressure condition? Would you the total pressure condition, would you expect the similar kind of results?
I don't think it is the pressure as such. It is the force that cells are applying on their the substrate on which they are. Because
cells as well.
Sorry.
Or the neighboring cells as well.
No, it is not on the neighboring cells. It is the what they are applying on the surface. So that's actually a good question. That if cells become too crowded, where they kind of start interact with their neighboring cells, then actually effect of the substrate kind of start to goes away. So it is not the cell or the interaction like with the neighboring cells. And if you talk about the total pressure, see, I mean, when you compare plastic with the gel, the total pressure is remaining constant. What is varying is the Young's modulus or the stiffness, and that actually in turn govern what amount of force the cells are applying on their surface. So that is the difference here. See, it is difficult for me to kind of speculate that, but as such, for say like for MSCs, people have shown that you can actually change their differentiation even by changing the pressure. For example, with higher pressure, MSCs try to go into more towards chondro, like the cartilage. So that people have shown. So I mean, I'm kind of sure that there will have some effect on pressure. But if you think from the practical angle, controlling the pressure of your cell culture system is much more difficult thing rather than just put them on a substrate and leave them like whatever they want to do.
Hi, Avijit. This is Shantanu here. Very interesting. Sorry.
Now probably you know what I want to come to you to discuss.
This is more of a curiosity. I know you have been interested in looking at telomerase activity and telomeres, and this is more just out of curiosity. Is it changing as you change the substrate? Do you see the length or activity of telomerase dependent on
exactly that is what I would like to come to you. My student came to your lab to learn the technique.
I know, but I didn't know that this was in this context. I'm sorry. So the other question is, I think you know Kundan's work from, he has shown that this chromosomes dependent on a particular lamine phosphorylation, the chromosomal distributions are dependent on the stress or the substrate, hardness of the substrate. So something like that going on here?
Okay. So answer one, I am in discussion with Kundan and we are thinking of having a collaboration together. Answer number two is that I have not shown that result, but if you grow them on plastic versus you grow them on culture, their lamine expression actually gets changed. So that, I mean, that result I have not shown here. So that is actually gives us a clue that nuclear rigidity actually gets modified, like when you culture them on plastic versus when you culture them on gel.
Abhijit here. Yeah, nice talk.
Thank you.
I'm just wondering, maybe you have done it. Did you look at the actomycin components after this?
Yes. So
because actomycin is known to play a havoc role in maintenance and differentiation.
Yes. So like I have shown you one result, I kind of quickly brushed through, is that when you culture the cells on plastic, say for 10 passage, versus you culture them on gel for 10 passage, then you measure their actomycin contractility separately, you find the cells which were grown on plastic has higher actomycin contractility compared to the cells grown on gel, even if now they are on the same substrate. Number one. Number two, we have checked for actin staining, and yes, you have higher F-actin the cells grown on plastic versus cells grown on gel.
But I would like to see, maybe you have done also PMLC staining, phospho squash staining.
Phosphorylation.
So that would tell you whether it's an active actomyosin or not. And the second issue, which is...
So, I mean, just, because the cellular contractility is different, it is...
No, but it would be nice to see.
Nice to see, obviously,
the
obviously.
Control.
I do agree.
And the other part is, very interesting part is your glioblastoma result. So there was one, I can't recollect now, I can tell you the reference later on. In an in vitro study, people have shown glioblastoma and actomycin interaction. GLI, not glioblastoma. GLI, I think that's a mutation which creates glioblastoma. So these two are known to interact in vitro to create aggressive tumor. So I thought you might like to put that axis in your
sure,
analysis.
that's a good. So in that particular work, I have not shown that. I mean, we are seeing that a change in EGFR expression, change in MMP secretion, depending on what kind of sickness.
One last question from Gautam.
Abhijit, there was some work from the Fredberg group a few years ago that looked at motion of cells in the presence of strain gradients. They find, I think they called it durotaxis or something.
Durotaxis.
So I was wondering whether that's the same reason why you say if you have an embedded sphere or cylinder below, you see this alignment and this motion in that
Exactly.
direction.
So we believe it so, and in fact, in any way, if you disturb the strain field, either you change your substrate from purely elastic to viscoelastic, you use inhibitor to inhibit actomyosin strain, you change the position of the bead, you change the composition. Any way you disrupt the strain field, you see that the structure is gone. So yes.
I still see some hands. I'm sorry, we will stop it here, and you can catch Abhijit during the lunch break. So lunch is served at Silver Oak, and the Jacaranda Hall at the first floor is also available. So it's same place where we went for tea. So see you back at 2:20. Please come back at 2:20 p.m. We will have further interesting topics to discuss.